les expresamos nuestro cordial saludo y bienvenida a este segundo día del desarrollo del sexto Simposio Internacional de Biología Celular y Molecular, año 2025. Bueno, es para nosotros un honor ser partícipes activos de este importante evento académico que está dirigido a estudiantes universitarios y profesionales a nivel nacional y también internacional, con una perspectiva que se enfoca en los nuevos paradigmas de la enseñanza, de la investigación formativa y también de la biología celular y molecular. El sexto Simposio Internacional de Biología Celular y Molecular, año 2025, tiene sus inicios en una continuidad de versiones anteriores que surgió como una propuesta en el contenido silábico del curso del mismo nombre, Biología Celular y Molecular, que año a año se imparte a los alumnos del sexto ciclo de la Escuela Profesional de Ciencias Biológicas, que junto a la plana docente hemos venido trabajando con mucho esmero, arduamente y de manera integrada durante el desarrollo de este semestre académico 2025-2 en la organización de este magno evento, persiguiendo como objetivos el ampliar la visión académica, ampliar la visión de investigación de todos los participantes, estudiantes, audiencia, tratando temas innovadores y de gran impacto en la biología celular y molecular, con un mejoramiento sostenido, persiguiendo el mejoramiento sostenido de la calidad de vida de las personas, el uso y manejo responsable de nuestros recursos de flora, fauna y recursos microbiológicos. También perseguimos como objetivo en el desarrollo de este evento el generar intercambio de conocimientos y estrechar lazos de comunicación por medio de este tipo de actividades académicas que son dictados por investigadores de amplia trayectoria académica y profesional. También con ello buscamos incentivar a todos los participantes a ser líderes en el campo de la investigación científica, con soporte a la creación y a la gestión del conocimiento, aplicación y también a la adaptación en cada una de sus áreas de competencia o en las áreas de competencia en las cuales estén interesados desarrollar en el futuro. Bueno, el evento lo hemos venido iniciando desde este día martes 2 de diciembre, en donde tuvimos el honor de recibir las ponencias de destacados investigadores con formación internacional. Por ejemplo, hablo del doctor José Morgado Díaz, exalumno de nuestra Facultad de Ciencias Biológicas, por allá en la década de los 80 aproximadamente, si no mal recuerdo, como él mismo lo contaba en la parte introductoria de su presentación. El doctor José Morgado es actualmente investigador senior del Instituto Nacional de Cáncer de Brasil y él nos compartió el tema sobre mecanismos de resistencia al tratamiento del cáncer. Nos habló sobre las vías de señalización involucradas en la población tumoral. El día martes también tuvimos el apoyo, la participación y el agrado de escuchar a la doctora María Paz Marzolo, quien es profesora investigadora, titular de la Facultad de Ciencias Biológicas de la Pontificia Universidad Católica de Chile, en Chile. Y ella nos estuvo hablando sobre la reelina, enfermedades del sistema nervioso central. Ella fijó los conocimientos sobre transducción de señales. Luego también tuvimos el amplio honor de escuchar a la doctora Irene Consuelo Julca Chávez, que también es una exalumna de nuestra Escuela Profesional de Ciencias Biológicas, exalumna nuestra, y con mucha satisfacción escuchamos su conferencia sobre el destino funcional de los genes duplicados. La doctora Irene es bióloga, como ya lo dije, formada en este claustro, la Universidad Nacional de Trujillo, en Perú, pero también sus estudios de posgrado lo realizó en España, en la Universidad de Granada, en la Universidad Autónoma de Barcelona, y actualmente es investigadora de la Universidad de Lausana, en Suiza. También, para satisfacción nuestra y como una evidencia de que nuestros alumnos pueden alcanzar sus metas académicas con formación internacional, tuvimos la participación también de otra exalumna nuestra, la doctora Karen Chauca Espinosa. Ella es investigadora postdoctoral en el Centro Nacional de Investigación Científica en Francia, en el Institut de Biologie de Paris, la CEN, en Francia, y doctora en Biología Celular y del Desarrollo por la Universidad de la Sorbona, en Francia. La doctora Karen nos habló sobre el desarrollo de las nuevas técnicas para estudiar la dinámica cortical de la actomiosina, desde el desarrollo de sondas ópticas hasta la microscopía de super resolución. Con ello, la doctora afianzó algunos de los temas que desarrollamos en el curso de en la cátedra, en las clases de la cátedra. Y el día martes cerramos con broche de oro las ponencias, teniendo el honor y el agrado de tenerlo como ponente al doctor Felipe Jiménez García. Quizá muchos de nosotros hemos leído su libro, que es de gran impacto a nivel de América Latina. El profesor Jiménez es profesor de carrera titular del Departamento de Biología Celular de la Facultad de Ciencias de la Universidad Nacional Autónoma de México. Él nos habló sobre los estudios de la estructura celular con el microscopio de fuerza atómica. Bien, y así habíamos culminado nuestra primera jornada de ponencias que se desarrolló el día martes 2 de diciembre. Con mucha satisfacción y entusiasmo vamos a iniciar este segundo día de jornada, jueves 4 de diciembre, recibiendo 4 ponencias, las cuales abrirá con broche de oro el reconocido investigador a nivel mundial, el doctor Harvey Lodish, quien es profesor emérito de biología en el Massachusetts Institute of Technology y es coautor del famoso libro Biología Celular y Molecular, que estoy segura la mayoría de nosotros hemos leído y lo hemos tenido como un libro muy importante en nuestra formación de biólogos y carreras afines a la salud y al estudio de la vida. El doctor Lodish, de un momento a las 10 de la mañana, nos estará hablando sobre las nuevas tecnologías y creación de empresas biotecnológicas exitosas para tratar enfermedades. Una historia personal. Ese es el título de su conferencia. Luego, continuando con las ponencias, tendremos el agrado de escuchar a la doctora Hilary Kohler, quien es una destacada investigadora en biología molecular, en biología celular, en biología del desarrollo, de la Universidad de California, Los Ángeles, en Estados Unidos de Norteamérica. Ella estará compartiendo con nosotros la charla intitulada Comprendiendo la base molecular de la quiescencia celular. Seguidamente tendremos también el placer de conocer y escuchar a Lionel Acosta Campos, quien él es un colega nuestro biólogo genetista, biotecnólogo, egresado de la Universidad Nacional Mayor de San Marcos, y tiene formación amplia en embriología, andrología, citogenética humana y biología molecular. Actualmente, Nacional es director del laboratorio del Centro de Fertilidad Fertigestar y del laboratorio de biología molecular en Genética Lab. Nacional nos estará compartiendo su ponencia sobre microARN en espermatozoides y su correlación con la reproducción asistida. Finalmente, para cerrar con broche de oro, valga la redundancia, esta mañana y también cerrar el simposio en su 6ª edición, tendremos el honor de escuchar a la doctora Bonnie Vittel, quien es investigadora principal del Instituto de Investigación Versity sobre sangre en Estados Unidos de Norteamérica. La doctora nos hablará sobre células B con baja expresión de inmunoglobulinas tipo D como estrategia terapéutica para aumentar la concentración endógena de células T reguladoras del tipo CD4 positivo FOXP3. Por tanto, estimada audiencia, estimados alumnos de la Cátedra de Biología Celular y Molecular, hoy tendremos un día muy productivo, de mucho aprendizaje, sobre todo ponencias que fijan clases teóricas que ya hemos venido escuchando durante el curso. Esta oportunidad, estimada audiencia, es valiosa porque obtendremos el conocimiento compartido por sus mismos protagonistas, que son los investigadores de reconocida trayectoria en la comunidad científica del mundo. Una vez más, quedan ustedes cordialmente invitados a dar continuidad al desarrollo del 6º Simposio Internacional de Biología Celular y Molecular, año 2025. Muchísimas gracias.
Muchas gracias, doctora. Agradecemos mucho la semblanza de la 6ª versión del Simposio Internacional de Biología Celular y Molecular. Agradecemos la participación de las autoridades que nos acompañan en esta mañana, así como a los reconocidos investigadores que nos honrarán con su participación como ponentes y a todo el público asistente que participará durante las diversas ponencias presentadas en este simposio. Al público que nos sigue en la transmisión en vivo por nuestra página oficial de YouTube y Facebook, les recordamos que el programa del evento está publicado en la página oficial de Facebook, tal y como lo podemos ver en estos momentos. Ahorita se va a compartir. Además, es de nuestro agrado informar que este evento es gratuito, de libre acceso y con emisión de certificado que evidenciará su participación. Para ser merecedores del certificado en mención, deben evidenciar su participación como asistentes, llenando por lo menos 7 de 9 formularios que corresponden a cada una de las ponencias. Se estará compartiendo los enlaces de estos formularios en un oportuno momento durante el desarrollo de cada una de las ponencias por nuestras páginas de Facebook y YouTube.
Good morning. On this occasion, we have the pleasure of introducing Dr. Harvey Franklin Lodish, who is a professor emeritus of biology at the Massachusetts Institute of Technology. Dr. Lodish will present his talk titled New Technologies and Building Successful Biotechnologies Company to Treat Disease, a Personal History. Dr. Lodish, welcome to this event. We appreciate your participation and support. Dr. Lodish, please remember that you have a maximum of 45 minutes for your presentation, after which we will have a 10 minutes question and answer session. We remind the audience in the room to keep their microphones and cameras off to avoid interruption during the presentation. Questions should be submitted via the chat during the talk and will be answered at the end. We also want to let you know that the link of the registration for link will be shared on your social media channels during the presentation. Please, Dr. Lodish, you may begin. We are listening.
There we go. Thank you very much. Can you see my screen?
Yes, Dr. Lodish.
Good. It's a pleasure to speak to this group. I will talk about the history of biotechnology, particularly building in Boston and Cambridge the largest collection of biotechnology companies and research in the world. My hope is that I will encourage some of the people in the audience in Peru to think about building their own biotech companies for therapies, diagnostics, devices, dealing perhaps with particular medical issues that affect your part of the world. At the end, I will give you my email address. I would be pleased to work with groups in Peru, as I do in many other countries, to try to help develop local infrastructure. To begin, I am an academic scientist. I've been on the MIT faculty and the Whitehead Institute faculty for over 50 years. I've trained 200 PhD and MD PhD students. I'm a real academic scientist. I've written textbooks on molecular cell biology. 2 of my students have Nobel Prizes. But like many of my colleagues at MIT, I am also an entrepreneur. I have helped start over a dozen biotech companies over many, many years. And I'll talk about 2, possibly 3 of them, depending on time: Genzyme, Tevard, and perhaps Cerberus. And then at the end, talk about work that I've done teaching biotechnology, the kind of course I would like to see incorporated at many institutions. Besides my academic work and founding companies, I've worked to build the industry in Massachusetts. Almost 20 years ago, I worked with the then governor of Massachusetts, Deval Patrick, to set up the Massachusetts Life Sciences Center and serve as the chair of their advisory board. This was a state investment over 15 years of $1.5 billion to build biotechnology, both in companies and to build many facilities and universities that would facilitate this research development. And I work closely not with the Secretary of Health or Education, but the Secretariat of Economic Development, because they realized, and I hope you realize, that building biotechnology will also create jobs and income for many people. Also, I have been privileged to serve on the board of trustees of Boston Children's Hospital. It's the leading pediatric research hospital in the world, and have helped them understand how to take their own research out of the hospital and build companies. A recent example would be the research there on sickle cell anemia that has led to the Vertex approved, FDA-approved gene therapy for sickle cell anemia. Comes directly out of Children's Hospital research. I also work with groups in many countries. Here you see me in Beijing, to work with not-for-profits that are working to develop treatments, diagnostics for rare diseases, diseases that affect many people, but usually in a specific ethnic group or location. And knowing little about the medical situation in Peru, I wouldn't be surprised if there were diseases that should be studied there. Now, biotechnology, as I'll explain, is only about 40 years old. But almost 60 years ago, I had the privilege of working in a chemistry lab at Stanford University. And Carl Gerasi, the head of the laboratory, I realized many years later, became my role model. He was, sadly passed away several years ago, but he was a very distinguished professor of organic chemistry, particularly steroid chemistry. And back in the 50s, he realized that starting from a chemical that he could isolate from a tree in Mexico, he could synthesize the progesterone derivative that became the first oral contraceptive, a chemical that was effective orally and far stronger than the natural hormone. He is known as the father of the oral contraceptive, the birth control pill. Importantly, he set up a company, Syntex, on the periphery of the Stanford campus, where they developed this first oral contraceptive and, of course, sold it. He became quite wealthy and used his money in many philanthropic endeavors. And I realized many years later, he was, in fact, my role model in thinking about biotechnology. So just a brief overview of biotechnology in Cambridge and a brief history of biotechnology and the importance of faculty entrepreneurs. So this is an aerial view of part of Cambridge, Massachusetts. You can see the Charles River in the background, Main Street, and on the right of Main Street is the MIT campus. The Whitehead Institute, where my lab was located, is on Main Street. And the whole surrounding area is filled with perhaps 200 companies that are developing drugs and therapies, including 18 of the world's largest pharmaceutical companies. This is just a few years ago, but this is the same aerial view about 50 years ago. You can see it was a cleared area. It was supposed to be a Kennedy Space Center, but when he was killed and Lyndon Johnson became president, they moved the Space Center to Houston, Texas. And what was left was a gift of open space that was used to build these various laboratories, companies, office buildings that house our biotechnology industry. Importantly, almost everything is within walking distance. This describes one of my companies, Rubius. I work at the Whitehead Institute, The company was founded by a venture firm, Flagship Ventures, which then was a 5-minute walk down the street. We started the company in an incubator that I will describe, a state-supported incubator for startup biotech companies. That was, again, a 5 or 10-minute walk in the opposite direction. When Rubius outgrew our incubator space, we've moved into commercial laboratory space that was at the other end of the campus. So I hope you come away with the notion that geography is important. Incubators are something that we have many of in Massachusetts. Some supported by universities, some supported by local governments. This one, the main one, is supported by state money, that is taxpayer money. If you walk in, you see a laboratory that would be appropriate in any modern research institution. What makes it different? It's a fully equipped laboratory. It has rooms, cold rooms, it has spectrophotometers, fax machines, DNA analysis machines, almost everything you need to run a modern laboratory. But what makes it different is that your company can rent a bench for $5,000 a month, and that's your company. In other words, it's co-working space. It's a not-for-profit incubator supported by the government that has birthed over 50 biotech companies that, again, like Rubius, leave the birthplace and are developed elsewhere. Lab Central also has a dozen or so private laboratories. So the company could expand from a few workers to perhaps 10. And as I said, this is the kind of structure that I encourage universities everywhere to build, perhaps on a smaller scale, to encourage the local development of biotech companies. A brief history of biotechnology, and I stress here the importance of faculty as entrepreneurs. As I pointed out, I have become an entrepreneur, but so have many, many of my faculty colleagues at MIT, both in biology and in the various engineering departments. And in fact, at MIT and many other US research universities, we are given 1 day a week outside professional activity, which means I can work as advisors to not-for-profit organizations, but I can also work with and consult for-profit companies, have shares in the companies and advise them. I can't be an operating officer, but I can certainly work with these companies. And we have clear conflict of interest rules. And to give you an idea, this is the founding group of a company many of you will know well, Moderna Therapeutics, that of course developed one of the major coronavirus vaccines. And there are many distinguished people on this list. I've highlighted the ones who are members of the National Academy. The head of the advisory board, Jack Szostak, had a Nobel Prize. You will see on this list people like Elizabeth Nabel, who is the president of one of the largest research hospitals in Boston, and many other distinguished people. The point is, it's the top scientists that get involved in these ventures. And this is what happened with the biotechnology industry. Until the 1970s, the word biotechnology was not in our vocabulary. And the revolution of recombinant DNA, the ability to clone a human gene and to use the clone gene to direct the synthesis of a recombinant protein. This was the birth of the biotechnology industry. Many companies were started in Cambridge, quite a number that I'm not showing here, like Amgen, Genentech on the West Coast. All of these started by faculty entrepreneurs who realized that this technology could make recombinant proteins that would be therapeutically useful. And these were and still are major products of the biotechnology industry. Proteins like insulin, growth hormone, clotting factors, which replace genetic defects. I'll talk about glucocerebrosidase, the Genzyme initial drug. Proteins like erythropoietin to stimulate red cell production used in kidney failure. And these built the initial biotech companies. And as you all know, there have been advances in molecular biology, cell biology, human genetics that have led to many other companies. We now have oligonucleotide therapies approved by the FDA, gene therapies, as I mentioned, sickle cell anemia, hemophilia, many others have been approved. And this is what drives us to develop more therapies. So let me give you an overview of what you need to do to start a biotech company. This is just an overview. I'm happy to go into details as you wish, and I'll talk about my course in a moment. But one needs several things. Some of them are scientific. For instance, intellectual property. You've developed a small molecule that could be potentially used to treat cancer or a genetic disease or whatever. That you have patents that give you protection so that someone else cannot practice your invention. You need freedom to operate. You need to know that no one else has a patent that can block you from carrying out your invention. You need a business plan. And I'll come back to the slide, but businesses are there not just to develop drugs, but to make money. Unless you can sell your product and earn money that can pay the investors who put money to help you develop the company, it's not going to work. So as I said, you need patent protection. You need freedom to operate. You need budgets. And you need to know who's going to buy your product and what the market size is. And what many of you realize is that these are not skills that you get in academic science. These are business skills. And in fact, the key to building a biotech company is having excellent science, but also working with colleagues who are expert in all of these areas: patent law, market research, research development, how you actually take a product. You can make a few milligrams of something in the laboratory. To make a kilogram requires an engineering skill that scientists don't have. And this requires a scientific advisory board and a board of directors. That is a board of directors that is the business board, that is responsible for raising money and actually building the company, and a scientific advisory board that would be comprised of leaders in the field that can advise the company about new developments, new technologies that could develop the research and so forth. And you'll see in a moment how this plays out in probably two of my companies that I'll talk about. And you need a helpful government and regulatory environment. So this is just a bird's eye view of what is required. And it's really a collaboration between scientists and business people. And at the end, I will talk about a course that I've developed and teach at MIT called The Science and Business of Biotechnology, where we bring together business school students and faculty and graduate students in the biological sciences and engineering disciplines to work together, culminating in writing business plans for startup companies. So let me give you a few ideas of what we have done. And I start with Genzyme, which, as I mentioned, was one of the first biotechnology companies ever. It started in the late 70s. And it was started by 8 faculty members, like myself, all at MIT. And as you can see, we were experts in many fields of biological science, but also engineering. For instance, bioprocess engineering, how you build and run bioreactors, biopolymers, molecular microbiology, and so forth. So it was a very good group of scientists. But Genzyme became well known for developing the first therapies for rare diseases. And that vision was not by the group of scientists, but rather by this gentleman who became a legend in biotechnology, Henry Termeer. He was the president. We hired him as president of Genzyme. He is not a scientist. He was a business person with an MBA. Henry understood the market. He knew that there were many rare diseases that affected under 200,000 people in the United States, and realized, we didn't, but he did, that one could build a company developing therapies for these rare diseases, selling them in such a way that one could build a profitable company. And as you may have noticed, eventually we sold Genzyme to Sanofi for $20 billion US. And the credit for building the company, sure, we as scientists had a lot of input, but it was really Henry. And I'll go into some detail for 2 reasons about our first product. And it was an enzyme replacement for a rare genetic disease called type 1 Gaucher. It's a lysosome storage disease where a particular enzyme called glucocerebrosidase, normally resident in lysosomes, is missing. And this causes the buildup of a glycolipid called glucocerebroside. In the most common form, type 1, it affects mainly macrophages. Types 2 and 3 are much more severe depending on the genetic mutation and can affect the central nervous system. But type 1 can be very debilitating. Bone marrow fills up with macrophages with undigested glycolipid, causing anemia, weak bones, many fractures. And like many genetic diseases, it's most prevalent in certain populations. In this case, it's in descendants of Eastern European, often called Ashkenazic Jews. Many mutations all spread in this population. And what we learned to do is manufacture the recombinant protein, inject it into patients in such a way that it would be targeted to macrophages. And we knew from work of others that macrophages had on their surface a receptor for polymers of the sugar mannose. Polymers of mannose occur on many fungi, other invaders. It's a pattern receptor. Here you can see a macrophage on the right binding to the surface of a yeast cell through its macrophage mannose receptors. And what we learned to do, in part based on research at NIH, is to manufacture the glucocerebrosidase as a secreted protein. And it would have on its surface several of these complex asparagine-linked oligosaccharides. And characteristically, these oligosaccharides have the sugars indicated in yellow, N-acetylneuromanic acid or sialic acid, galactose, and acetylglucosamine. But what we did was manufacture the protein with these sugars and then treat the protein with the three enzymes indicated on the right, neuraminidase, beta-galactosidase, and acetylglucosaminidase, leaving a truncated oligosaccharide that has three exposed mannose residues. And we showed that this was sufficient when we injected the recombinant protein into a patient, that it would go to macrophages and be targeted to lysosomes and cure the disease. And again, you don't need to be a clinician to see the effect of enzyme replacement therapy. Here you see this young boy with a hugely enlarged spleen and liver characteristic of the disease. And within a few months of enzyme replacement treatment, he's fine. So you begin to see how this drug works and the fact that we used it to build Genzyme as a company. There's also a very personal reason for mentioning this, in that many years after I left Genzyme, we're talking 2002, we realized that one of my grandchildren was going to be born with Gaucher disease. It turns out our daughter and her husband are both carriers of Gaucher, and our daughter had amniocentesis done on the child she was carrying. Andrew was, we knew that the child would be born with Gaucher, with type 1 Gaucher, and could be treated with the Genzyme drug. And here you see my grandson. He's now 23. When he was 10 years old, being transfused with the Genzyme drug. And I'm pleased to report that he has been privileged to lead a completely normal life. He was on the Genzyme drug for 12 years. He's now on a pill that blocks the synthesis of the glucocerebroside. Here you see him bicycling across the United States as he did one summer recently. But more importantly, Genzyme used the technology that we had developed to develop enzyme replacements for several other rare diseases. And as I said, we built the company around developing drugs for rare disease. And again, when I say we, I'm talking about the 8 of us who put the company together that really served as an advisory board to help build the company for the first years of its existence. Let me talk briefly, I won't be able to get to Cerberus, but I'll talk briefly about TEBARD and then the course that I teach. And of course, I will take questions. Tevard is a company I started with two business people, Daniel Fisher and Warren Lammert, who have children with a devastating neurodevelopmental disease called Dravet disease. And the name Tevard is Dravet spelled backwards. The hope was we started the company to reverse Dravet. Here you see Daniel's twins. The one on the left is afflicted with Dravet. The one on the right is well above average, quite normal. And just to give you a bit of the background, it's a haploinsufficient genetic disease caused during the child's development of loss of one copy of a gene. In this case, the gene is SCN1A, which encodes a particular voltage-gated sodium channel that's important for inhibitory interneuron function in the brain. And because there's only one copy of the gene, insufficient amounts of this voltage-gated sodium channel are made, and therefore the neurons malfunction. And we started developing a novel therapy for the disease. What we realized is that many inherited genetic diseases are caused, regardless of the disease, are caused by nonsense mutations. As you know, there are 61 codons that encode amino acids and 3 so-called stop codons, UAA, UAG, UGA, that are normally found at the 3' end of messenger RNAs. But depending on the gene, around a quarter of all human genetic mutations are caused by a change of an amino acid codon to one of the 3 stop codons. And this, of course, makes a truncated protein that is non-functional. And that was the case, as it were, of the 2 daughters. And I'll save you many, many years of development, but we developed tRNAs that have many mutations, including an anticodon that allows them to recognize one of the 3 nonsense codons, TAA, TAG, TGA, and put in the correct amino acid to make the full-length protein. As it happens, a major type of human genetic disease is caused by an arginine to a TGA mutation. This is the case in the 2 girls. And here you see a cell culture experiment where we express the normal copy of the SCN1A channel. It's a very large protein, and you can see the protein on the blot on the left. If you inject the mutant protein, you see nothing because you make a fragment of the protein and it's degraded. But if you express the mutant protein, and we use an AAV vector to express the gene encoding the suppressor tRNA, you make the full-length protein, and as expected, the full-length protein has normal function. And this works in a mouse. Here you see a mouse with the SCN1A mutation. As the black curve indicates, the mouse dies. It survives just a few weeks, and then it's moribund. But by expressing our AAV suppressor tRNA, we put in the normal amino acid, the mouse is rescued and lives essentially a normal life. Yet we decided that as a company, that we would start not with Dravet syndrome, but start with other diseases that don't affect the central nervous system, that have, as it were, a larger population. And the ones we are currently working with are Duchenne muscular dystrophy, and particularly a heart disease, Titin dilated cardiomyopathy, which are caused in many cases by nonsense mutations. And as you may know, Duchenne muscular dystrophy affects this very large protein called dystrophin, which functions in all muscle, including heart, to connect the actin cytoskeleton, the contractile apparatus, with the cell membrane. And in its absence, the muscle cell gets ripped apart, and that's what causes the death of these childs. It's an X-linked gene, so it mainly affects boys who have only one copy of the dystrophin gene, and that would get inactivated. As I said, it's an enormous protein. The protein is over 3,600 amino acids. There are therapies that make microdystrophins. And although many of these have been approved, they actually don't work. You need all of these 24 spectrin repeats that are the springs that keep the protein functional. To get to the point, we can use our AAV vector here in a dystrophin mouse and make enough of the dystrophin protein to rescue the lethality of this MDX mutation, the dystrophin mutation. I'll spare you all of the details, but in this experiment, we can make as much as 20% of the normal level, which is sufficient to rescue the genetic disease. We actually have much better tRNAs, and we're now up to close to 100%. And as you can see, the dystrophin is expressed in almost all of the muscle cells because the AAV suppressor tRNA rescues the nonsense mutation in virtually all of the cells. And most importantly, by physiologic measurements, such as how long a mouse can hold on to a rotating bar called the rotarod. They're almost up to the wild type level, but enough to survive. And again, we are doing the same thing for Titin and hope to be in clinical trials next year. So I'm going to skip over Cerberus and talk briefly in the last 2 or 3 minutes about courses that teach biotechnology. I've been teaching biotechnology courses at MIT for over 40 years. I actually taught the first biotech course in the world with a professor of chemical engineering back in 1984. The current course brings together students in the School of Engineering and the life sciences, the departments of biology, together with MBA students in the Sloan School of Management, the business school. And the idea is to give them all insights into what is currently going on in biotechnology, both in the science and in the business. So we have classes in which everyone attends on Thursday, in which we go through, you can look at the list, many current topics in biotechnology. Therapeutic proteins, gene therapies, nucleic acid therapies, monoclonal antibodies, cancer immunotherapy, vaccines, neurologic disorders, small molecule drugs, new kind of molecules called PROTACs. But we also bring in outside speakers to talk about many of the practical aspects of being a company. how technology licensing offices at universities function to license technology from the university into companies, patents, starting and building biotech companies, how you execute a clinical trial, how you get drug approval by the FDA and regulatory agencies in other countries. Give you an idea of many of the people that we bring in as speakers. You can see representatives, Tony Ho, Amy Schulman, Bob Tepper, from many of the leading life science venture firms, a number of faculty members who have started and built companies, people like John Maraganori, who built Alnylam into a multi-billion-dollar company. And then we have recitation sections. On Tuesday, we have a Sloan MBA giving a crash course in biotechnology business, investor pitches, fundraising, valuation. And we also have a biology PhD who gives a crash course in molecular biology and immunology to the business students. And the whole idea is that they get together, the business students, the science and engineering students, and work together to write a business plan for a biotech startup that they submit as a research paper, but also a presentation. So this is one of the signature courses that we use to build biotechnology and train students in Boston. And importantly, a version of this course several years ago has become an MIT edX course. It's online. Whoops. It's online. Any of you can go in and take the course. It's free. It's had over 34,000 students, which I believe is a huge number of students. You also see on this slide my email if any of you want to get in touch with me. So I'm done. I hope you've learned something from this. I'm stopping to share, and I'm happy to take questions. So whoever's in charge in Peru, get me questions. Julia, hi.
We thank Dr. Lodish. Thank you very much for your presentation. Now, Dr. Llanos will proceed to deliver the question from the audience.
Good morning, Dr. Lodish. It's exciting to have you here. Thank you so much for your excellent talk. It is an honor. And I send my greetings in behalf of the Professor Fatima and all the organizers for your presence. So I will proceed to deliver the questions from the audience. So please let me check. So first question, in your opinion, what is the most common mistake scientists, founders, including yourself in the beginning, make when assembling and start? Oh, do you have problems with?
I'm having trouble hearing you. You come.
My apologies. Okay, now it's better, or is it still fine?
It's better now. Yes.
Okay, I will formulate the question again, please. Thank you. Okay, in your opinion, what is the most common mistake scientists, founders, including yourself, in beginning, make when assembling a startup's initial leadership team? Do you look for anything specific in a CEO or CSO that goes beyond the resume?
Well, the major mistake people make is not thinking of companies as a business. And many scientists academic scientists try to run their own companies. I've never run a company. I would not be good at it, you see. I always work with people who have some science background, but really understand business. They understand finance. They understand human resources. They understand manufacturing, you see. These would be engineers. In other words, to build a company, you need people with many different expertises. And most scientists don't realize this. This is why we teach that course that I described, that you really need to assemble a group that you can work with. It has to be a collaborative group, but with different backgrounds and expertises. So for instance, the chief scientific officer, and you need such a person when you have 20 or 30 people in the company. That person should have a broad view of science, but also enough business expertise to be able to explain the science to investors, you see. That's a skill that I've learned. Conversely, the CEO is often a business person, like I mentioned Henry Termeer. You see, I mentioned Terrar, Daniel Fisher is the CEO. He is not a scientist. He's an electrical engineer, but has an MBA. So that's a long answer to the question, but it's a collaborative project and you need to bring in people with many different backgrounds.
Okay, we understand. Okay, I have another question from the audience. So, okay, I'm going to deliver it. Dr. Lodish, thank you for your presentation. I would like to ask you about the role that omics biotechnology play today. Have you heard about the recent discussion around quantum biotechnology?
I'm not familiar with quantum biotechnology, but these are techniques. These are not ends in themselves. These are techniques. Artificial intelligence, which you may ask me about, is a technique. And it's aimed at developing drugs, therapeutics, and so forth. So keep that in mind. These are not aims in themselves. These are techniques that are constantly evolving to help us do the job better.
Okay, Dr. Lodish, I will follow with the next questions. It seems everybody's excited to ask you questions. So please let me follow. Okay, so I have another question. So considering the cost of treatment of rare disease, how expensive it is to treat rare disease using biotechnological products?
Well, you have to think of the cost of not treating the disease. You say, Gaucher disease as one example. The children are in hospitals a lot. And yes, the therapy is expensive, but it allows children to live a normal life. Similarly, we've had this discussion, for instance, the gene therapy for sickle cell anemia, which is a horrible genetic disease. And the gene therapy is given to the most serious patients with the disease who have required multiple hospitalizations, you see. So it actually becomes cost effective. Plus, it allows them to live a normal life. But I agree, these are expensive technologies. On the other hand, as competition increases, we hope that they will be less expensive. You see, this is a dilemma. And in countries that don't have high-level medical systems, it becomes a problem. On the other hand, as I point out, for rare diseases, which are confined generally to specific ethnic groups, the way to start is prenatal testing. You see, the real way to treat these inherited diseases is prenatal testing. And an example that I often use are these so-called Jewish genetic diseases like Gaucher and Tay-Sachs. And strikingly, about 20 years ago, a religious leader, a rabbi in the Orthodox community in Brooklyn, New York, established a screening protocol for every young person in the community as they go through adolescence. So what happens now is each child, as they go through adolescence, knows their genetic profile. And couples, eventually, if they're serious about marriage, they will exchange this genetic information, you see. But what they've done is eliminated many of these diseases in one generation. And that's really the way to go. And if there are such diseases, and there are 7,000 of them, and we don't know the basis of most of them, you see, this is a much more economic way of treating the disease. I was actually talking to a young student at MIT who is part of this community, this religious Orthodox Jewish community. And she tells me before they even go on a date, they exchange genetic information. It sounds crazy, but it works, you see, and it empowers the young people, you see, to make these decisions. So yes, enzyme replacements and so forth are a solution, but you want to take the long view. So when I get in these discussions, I encourage companies, I encourage governments to really support prenatal testing. It's much cheaper.
Okay. Oh, I guess I would like to do that if I get married somehow. Okay,
Well,
it would be exciting.
This is the way to go. And again, this is done usually not through physicians, but through religious leaders, cultural leaders, imams. I know this is done in several of the Muslim communities in the Middle East. You take advantage of the community leaders to organize this, and it can be very effective. Sickle cell anemia the same way. Beta thalassemia the same. More questions? We have time?
Yes, we have time. Thank you. Dr. Lodish, what advice would you give to the young students who are listening us right now? And would you like to start a biotechnology company in the future?
Well, I would encourage young students who think they might be entrepreneurs is to learn. And, ideally work in a biotech company somewhere. Many MIT students will graduate as undergraduates and work in companies for 2 or 3 years. Then go to graduate school and get an advanced degree. But many of them will stay in the industry and eventually go on and get an MBA. So unfortunately, our current government in the United States makes it difficult for immigrants to come in and do that. But there are many other countries that people could go to work in biotech companies and just experience it. And possibly even as undergraduates, if you can, to take business courses as part of your curriculum. You know, science students take courses in literature and government and economics, and there's no reason they can't. Many MIT students do this. You see, so I'd encourage young people to think about this, but how you could build a company that would serve a need in your community, in your country. In other words, is it a genetic disease? Is it a diagnostic that needs to be done? You know, setting up a genome sequencing company for cancer. You know, a company that would locally work with hospitals to sequence the DNA of tumor cells to advise oncologists how to treat the patient. That sort of thing. Doesn't have to be highly innovative, but there are many local companies that could play a huge service by just thinking what the needs are of the communities, the medical communities, and so forth in your communities. And this is not just Peru. This is advice I give. I mean, a month ago, I was in Tashkent, Uzbekistan, and Almaty, Kazakhstan, giving the same talk.
Okay, we have another question. Time is still helping us, cooperating. So from your experience, co-founding, Genzyme, and others, what was the most counterintuitive lesson you learned about translating basic cell biology discoveries into core of a viable company? Has a project that seemed elegant in science ever turned out to be impractical as a business, and vice versa?
Exactly. Again, these are businesses. I often start my lectures by saying, businesses have to make money. There is a lag period. I didn't have the opportunity to go into detail, but it takes many years to develop a drug. It takes fewer years to develop a diagnostic, but you need investors who will trust you with their money so you can carry out the research, but they will always look for a return on their investment. And that's a mental state that you have to think about. And it's uncomfortable for many scientists because they think they have a very elegant solution to a disease. But when they realize, yeah, I have a gene therapy for cancer, but I can't deliver it to the tumor. You see, it's questions like that, you see, or it's impractical to manufacture. So you have to think of the whole pattern from the scientific discovery all the way to the clinic. What would you actually use to treat patients? What would it be, you say? How are you going to deliver it? You know,
Okay,
and
Dr. Lavish, thank you.
yeah, I'm not hearing you, Maria.
Thank
I see you, but
you so
you're there you go.
Okay, So we are very grateful for your time. And now we are going to continue with the moderator to
Yeah, again, I can't hear you.
Julia. Oh, please, Julia. Thank you very much, Dr. Lodish. Thank you.
Thank you all. And again, if any of you have specific questions, please write me. I do try to respond to my email. Okay?
We thank Dr. Lodish for his excellent presentation, which I am sure has been of great benefit to everyone. Thank you for sharing your knowledge and experience in cell and molecular biology. We also thank this morning's participants for their attendance. We invite all attendees to join us for the presentation of our next speaker. Thanks.
Thank you. I will sign off.
Good morning. It is our pleasure to introduce Dr. Hilary Kohler, professor at the University of California, Los Angeles, USA. Today, Dr. Kohler will deliver her talk titled Understanding the Molecular Basis of Cellular Questions. Dr. Kohler, welcome to this event. We thank you for your participation and support. Dr. Kohler, please remember that you have a maximum of 45 minutes to deliver your lecture, followed by a Q&A session lasting up to 10 minutes. We kindly ask all attendees to keep microphones and cameras turned off to avoid interruptions during the presentation. Please submit your questions through the chat during the talk. They will be addressed at the end of the lecture. Additionally, we inform you that during the presentation, the link to the attendance form will be shared through our social media channels. Please proceed, Dr. Kohler. We are listening.
Great. Good morning. Thank you. I'm delighted to be here and so happy to have this opportunity to talk with you and tell you about some of the research in my laboratory. This is really a pleasure for me. So thank you so much for the opportunity to talk with you this morning. I really appreciate it. If I do this, can you see my screen?
We were watching your screen before.
You were? Okay, let me go back then.
Perfect. Thank you.
Can you see it now?
Yes, great. Thank you.
Great. Great. Well, thank you again so much for the opportunity. I'm delighted to be here. My name is Hilary Kohler, and I'm a professor at the University of California, Los Angeles. And it's really my pleasure to tell you about some of the research going on in my laboratory. So many of the cells in our body have an important decision to make every minute of every day. They need to decide whether to replicate their DNA and continue through the proliferative cell cycle, or to exit the proliferative cell cycle and enter a reversible state of quiescence. So we think that quiescent cells are extremely important for understanding normal physiology and pathology, and yet we as a field have a very poor understanding of what it means to be a quiescent cell. So when a quiescent cell is activated to proliferate, usually that's a sign that there's some sort of emergency. For instance, there's a wound that needs to be healed, or an antigen that needs to be responded to. And so we in my laboratory think that understanding the molecular basis of quiescence is really critical for understanding human physiology and pathology. So one more question. Can you see my cursor if I move it?
Yes, doctor.
Yes, you can see it. Okay. So many different types of cells enter a similar quiescent state. So here are yeast, and yeast enter a state that's very similar to quiescence in human cells. And yeast will enter a quiescent state, for instance, if they're nutrient deprived. C. elegans have cells that enter a quiescent state, and the C. elegans entire organism can enter a state that's kind of similar to quiescence. And within our bodies, there are different types of cells that can become quiescent. So T cells, as you see here, and importantly, many different types of stem cells, including a muscle stem cell shown here, can enter a reversible state in which it's not dividing at any particular time, but has the capacity to come back and proliferate again. So in my opinion, quiescence has been very important for the presence of life on all different parts of the planet, because quiescence is really important for the ability of cells to survive in times when nutrients are limited or the environment is inhospitable. And so for that reason, I think quiescence is a really important state. So here are just some conditions in which quiescence is really important. So for instance, the context of wound healing. So if there's a wound, it's important for cells to leave a quiescent state, start to proliferate and heal the wound. The context of tissue regeneration, for instance, if there's damage to a tissue or you need to repair a muscle or the liver, cells that are quiescent will start to proliferate and regenerate that tissue. And nutrient-limited conditions, so for instance, in inhospitable environments or places where there aren't a lot of nutrients, the ability of cells to exit the proliferative cell cycle and retain viability, retain their capacity to come back and proliferate is really important. In my opinion, there are a lot of pathological conditions that also have quiescence at their fundamental level. So cancer, as we know, is a state where cells continue to proliferate, even though they're supposed to be quiescent. In the case of autoimmune disease, there will be immune cells that ought to exit the proliferative cell cycle but continue to proliferate inappropriately. In the case of tumor dormancy, those are cells that are part of a tumor that will become quiescent or dormant, sometimes for long periods of time, but then can come back, proliferate, and lead to the tumor coming back. And finally, chronic wounds. So chronic wounds are cases where wounds don't heal during the normal progression and instead fail to heal. And the failure to heal of chronic wounds, which is actually a very common problem, can reflect the fact that cells that ought to have been properly quiescent and ought to have the capacity to come back, proliferate, and heal the wound, maybe were not in the proper quiescent state, and as a consequence, don't have the capacity to come back, heal the wound when they proliferate. So many different types of signals can converge on kind of what we think of as a common quiescence program. So in response to things like biomechanical cues, such as changes in stress fibers, changes in cell shape, an absence of nutrients, a lack of mitogens, which are signals that tell a cell to proliferate, in response to contact inhibition, so a sense that a cell has other cells that are touching it, in response to extracellular matrix and integrins, the extracellular matrix proteins can give this signal. And in some cases, even within cancer therapies, all of these different signals can tell a proliferating cell that it's time to be quiescent, that it's time to exit the proliferative cell cycle reversibly with the capacity to come back and proliferate again. And when all of these different types of signals converge on quiescent cells, the quiescent cells in different organisms and response to different signals tend to have similar morphological, gene expression, and functional characteristics. So some of the morphological changes that tend to happen are that the cells tend to become smaller, they tend to compact their chromatin, their DNA. They tend to undergo changes in cell cycle expression genes, so they downregulate cell cycle genes, and they'll upregulate kind of quiescence genes. They'll have other functional changes, such as a resistance to stress, which can be valuable, for instance, if you're nutrient deprived. And in our lab, we've been very interested in the fact that form follows function, and we wanted to know, do the sizes and shapes of cells actually affect their functional roles? So what is it about the size and shape of quiescent cells that maybe is important for its characteristics and its functions? So in our laboratory, one of the models that we've relied on heavily for quiescence is in fibroblasts. So fibroblasts are cells that are in lots of different tissues of our body, including your skin, and they have the important job of secreting extracellular matrix proteins. So they were first identified by a German physiologist, Rudolf Virchow, which is shown here, and he called them spindle-shaped cells in the connective tissue. And so you can see some really nice early drawings of fibroblasts here. And fibroblasts are really crucial for both physiology and pathophysiology. So they secrete extracellular matrix proteins. So you can see some fibroblasts here in an epithelial lining with the epithelial lining over here. And so they will secrete the extracellular matrix proteins that give our tissues strength and elasticity and resilience. In the context of wound healing, fibroblasts are very important for orchestrating the healing of the wound. And fibroblasts can go rogue. They can contribute to disorders. So for instance, here you see keloids, and that's a case where at the end of the wound healing process, the fibroblasts continue to proliferate. So in our laboratory, we have a number of questions about quiescence that we're trying to address. So we would like to understand what are the cellular changes that occur as cells become quiescent, and what is their molecular basis. We'd like to understand how do distinct quiescent signals converge on common morphological changes and gene expression changes. We'd love to know what regulates the gene expression changes that occur with quiescence. And we'd like to know, are the gene expression changes with quiescence actually related to the functional attributes of quiescent cells, both in vitro and in vivo? And we believe that if we have a better molecular understanding of quiescence, that this will hold promise for us as people who try to not only do basic research, but also help the world. So our hope is that an understanding of the molecular basis for this proliferation quiescence transition will allow us to manipulate it so that we can shift cells into and out of quiescence therapeutically. And our hope is that by doing this, we would be able to help patients with diseases. So for instance, could we activate quiescent cells to proliferate and thereby enhance regeneration and healing? Could we activate the dormant cancer cells and make them more sensitive, for instance, to chemotherapy? Could we shift proliferating cells into quiescence to reduce inappropriate proliferation, for instance, in the context of keloids, autoimmune disease, or cancer? So in our laboratory, we've been using a fibroblast quiescence model. We get fibroblasts that are shipped to us from skin that's shipped to us straight from patients, normal skin. And these primary human cells, we can grow them in culture. We don't need to immortalize them in any way. So we don't need to inactivate tumor suppressors or activate oncogenes. They just grow in culture. We grow them for a limited period of time, and then we're done with them because eventually they'll start to run out of replicative lifespan and senesce. And we can use two independent quiescent signals in order to make the cells quiescent. One of them that's shown here is we can serum starve the cells. So we remove the serum, which contains mitogens, and then the cells will enter a quiescent state. And the other that's shown here is that the cells can become contact inhibited. So they'll start to touch each other. For either of these two signals, we think that they're physiologically relevant because, as I told you, fibroblasts are very important for wound healing. And we think in the context of a wound, the cells will receive signals from serum telling them that there are mitogens nearby, and they'll have more space to grow. So we think that the fibroblasts that are quiescent and, for instance, normal skin likely are getting signals similar to the ones that we're providing to our cells in culture. And the other really nice thing about this model system is that we can make the cells quiescent by either of two quite distinct signals, and we can compare. And that way we'll be able to see, are any changes that we see just a consequence of, for instance, removal of serum or cells touching each other, or do they really have to do with being quiescent? So if we see them in both situations, that will suggest to us that they're kind of essential for quiescence. The other really nice thing is that we can reverse the signals, and that way we can tell, is it just that the cells have been in culture for longer, or did we really have a reversible change with quiescence? So one of the approaches that we've taken to try to understand quiescent cells is to do RNA sequencing. So for this, we had proliferating cells. We made them quiescent either by depriving them of serum or by contact inhibiting them with full serum. And then we took those serum start cells or contact inhibited cells, and we either gave them back serum or we replated them at a lower density, and that allowed us to reverse the quiescence. And then we captured all of these samples and did RNA-seq on them. And what you can see from this heat map is that thousands of genes actually change with quiescence. Some of these genes were higher in the proliferating cells, lower in the quiescent cells, and then come back with a restimulation. And others were lower in the proliferating cells, higher in the quiescent cells, and then go back down when the cells are restimulated. The other thing that you can learn from this heat map is that the changes with contact inhibition, the changes with serum starvation, even though they're two very distinct signals, actually look very similar. And so this reinforces our finding that there is a common and reversible gene expression program associated with quiescence. So one of the things our laboratory is very interested in is trying to understand what is it that regulates these gene expression changes with quiescence. So it's known, as I mentioned, that there are global changes in chromatin with quiescence. So this is very clear in T cells. So in T cells, you can see that they will go from a very compact state to a much more open state. This is the nuclei when the cells become activated. And we looked in our cells, and we could say that when we compared proliferating cells, contact inhibited cells, and restimulated cells using dual color fluorescence in situ hybridization to get an understanding for how big or how compact or expanded chromosome 16 is, we could see that in our model system as well, when the cells were contact inhibited, the chromatin shrunk, and when they were restimulated, the chromatin got bigger again. So we hypothesized that perhaps histone post-translational modifications are important for quiescence, for these chromatin conformation changes with quiescence, and for gene expression with quiescence. So DNA is wrapped around an octamer of histones, and those histones have tails. Those tails get modified, and what we wanted to understand was whether these modifications might be characteristic of quiescence and might be actually functionally important for quiescence. So in order to test this, in collaboration with our colleague Ben Garcia, we isolated histones from proliferating and quiescent cells. We used trypsin to digest them, and then we used mass spectrometry to monitor the levels of different histone post-translational modifications. And what we learned was that among the changes in histone post-translational modifications between 14-day contact inhibited versus proliferating fibroblasts, we saw that surprisingly there were relatively few histone modifications that changed. I actually thought that there would be more, but we saw big changes in one particular histone modification. So histone H4 lysine 20 can have no modifications, one modification, two modifications, or three modifications. And we found that the trimethylated form really increased when the cells became quiescent compared with when they were proliferating. And so here you can see no methyl group, one methyl group, two methyl group, or three methyl group. And you can see there's a protein called KMT5B or 5C. So these are pair of proteins, 2,4,20H2 or KMT5B and C. And they put these methyl groups on. And here we've used immunoblotting with an antibody against H4K20 trimethyl. And we looked at proliferating cells, cells in G1, cells that were contact inhibited for 14 days or 21 days. And we could see H4K20 trimethyl increased when the cells were contact inhibited. And here we could see that they were proliferating, they were contact inhibited, and then we re-stimulated them for 48 hours, and we could see that levels of H4K20 trimethyl reduced. So the levels of this particular histone modification increase when the cells become quiescent, and it does so in a reversible manner. And we've used immunofluorescence. We looked at proliferating cells, contact inhibited cells, and serum start cells. And we can see that H4K20 trimethyl levels increase when the cells become quiescent. In addition, when we look at our RNA-seq data, we could see that SUFR20H2, which is one of the histone methyl transferases that can create the H4K20 trimethyl mod, was upregulated with quiescence. So these are all the genes involved with chromatin remodeling, and SUFR20H2 was upregulated. So we used a technique called cut and tag, which would tell us where in the genome the H4K20 trimethyl marks are. And we could see that H4K20 trimethyl was more abundant in the contact-inhibited cells. So this is the tracks for the proliferating cells. Here are the tracks for contact-inhibited cells. We could see that it was more abundant in the quiescent cells, and it tended to localize to promoters, to gene bodies, and what had been reported before, which are repetitive genomic elements. So H4K20 trimethyl had been associated with chromatin compaction, and it had been previously reported to be at centromeres, telomeres, transposable elements. In our case, we were curious, so all the gene expression changes that occur with quiescence, could it be that H4K20 trimethyl was sitting at the promoters? So we looked and we saw that among the genes with H4K20 trimethyl and peaks in their promoters in quiescent fibroblasts, about 5% were upregulated with quiescence and 5% were downregulated with quiescence. So we concluded that H4K20 trimethyl just sitting at promoters was unlikely to be kind of the reason why genes were being regulated with quiescence. So we took kind of an unbiased look and we asked, okay, if we take a look at the H4K20 trimethyl peaks and we ask where do these overlap with ChIP-seq data sets that other people have done before, we could see that there was an enrichment for SMC1, SMC3, and CTCF. So these are cohesins and CTCF. And cohesins and CTCF are very important for marking the boundaries between chromatin topologically associating domains or TADs. So we know that the chromatin in our nuclei is associated in forms like big kind of circular complexes. And these are called topologically associating domains. And the boundaries between these are marked by CTCF and by cohesin. And so this suggested to us that perhaps H4K20 trimethyl is also something that marks the boundaries between these big topologically associated domains. So in order to look at this more closely, we looked at previously published Hi-C data. So Hi-C is an approach where you can look at how much different portions of the genome interact. And if you see triangles in the Hi-C data, that will show you that that's a topologically associating domain, and those regions tend to interact with each other. Whereas here's another topologically associating domain, and the DNA in those regions tend to interact with each other. It tends to be that genes that are regulated by specific promoters, those genes and their promoters tend to be within the same topologically associating domain, and it's thought that that's an efficient way for enhancers to interact with particular genes. So we looked to see where H4K20 trimethyl was in comparison to this Hi-C data, and we could see that, at least in some portions of the genome, it was clear that these, for instance, each of these triangles looks like a topologically associating domain is in a section where all of this chromatin tends to interact with each other. And H4K20 trimethyl, our peaks shown in turquoise here, tended to be at the boundaries between these domains. And you can see that when the cells were contact inhibited, we had much bigger peaks. So we had much more H4K20 trimethyl there at these kind of insulators, the boundary marks between the TADs. So we already know that CTCF tends to bind at boundary marks between TADs. And so we asked, is H4K20 trimethyl enriched at the same sites that can have CTCF binding? And so when we looked for the consensus CTCF binding motif and looked to see across all CTCF binding motifs, what's the distribution of H4K20 trimethyl in our cut and tag data, we could see a clear peak. And so it looked as though, yes, H4K20 trimethyl can bind to the same place that we have consensus CTCF binding motifs. And so this suggested to us that maybe there's some sort of association between CTCF and H4K20 trimethyl. And we started to wonder, well, if they can localize to the same places, do they cooperate? Do they compete? And so we looked at our cut and tag data for H4K20 trimethyl, and we generated cut and tag data for CTCF, and we compared. And what we could see was that there's specific portions of the genome in which CTCF levels are high in proliferating cells and are reduced in contact inhibited and serum starved cells. And so you can see all these regions here. And so they have like strong CTCF peaks when the cells are proliferating, and the levels go down a lot with quiescence. And we found that there were 1,149 such regions in the genome with peaks that were lost with quiescence. And when we applied the same filters, we found only 14 peaks that had actually become more, where CTCF had become more abundant in the quiescent cells. And then we asked, okay, across all of these peaks, what happens to H4K20 trimethyl? So if we looked at all CTCF binding sites, we didn't really see much change with H4K20 trimethyl. But if we look specifically at the sites where CTCF became less abundant, when the cells became quiescent, we could see a clear peak of H4K20 trimethyl at these sites. So given that CTCF peaks are being reduced and H4K20 trimethyl is coming, this suggested us that perhaps there's some sort of antagonistic relationship between the two. And so one of the things we wanted to ask is, okay, well, is CTCF actually downregulated with quiescence? And so we used immunoblotting, and we saw that, yes, CTCF in whole cell extracts, and that the chromatin becomes less abundant when the cells become quiescent. And this was true both for contact inhibited and serum start cells. And we saw this by immunofluorescence too, that when the cells became quiescent, by contact inhibition or serum starvation, CTCF levels declined. When they were restimulated, we could see CTCF levels came back. So then we looked across, this is one particular chromosome, and we asked, okay, how do the CTCF peaks and the H4K20 trimethyl peaks relate to each other? And what we could see was that there are portions of the genome that have tons of CTCF, as you see here, but then there are other portions of the genome that seem to have a lot of H4K20 trimethyl and a lot less CTCF. And this seemed to be kind of a pattern that we were observing, that different portions of the genome seem to have either more CTCF or more H4K20 trimethyl. And when the cells were proliferating, so here's CTCF in proliferating cells, we thought that we were seeing more CTCF, so the CTCF seemed to get more intense, whereas when the cells were quiescent, the regions didn't change, but the H4K20 trimethyl rich regions became even more H4K20 trimethyl rich when the cells were quiescent. And we also approached this using a complementary approach of super resolution microscopy. So here we've stained for H4K20 trimethyl in green and for CTCF in purple, and we're getting a very clear view of the nuclei of the cells, and we could see that there seemed to be distinct territories. So the serum start cells had a lot of the green H4K20 trimethyl kind of foci, and some but fewer CTCF-rich foci, whereas in the proliferating cells, we saw some H4K20 trimethyl foci and more CTCF foci in the purple. And it also looked to us like the CTCF foci were kind of bigger and fluffier, and the H4K20 trimethyl foci were like more compact and smaller. And we've done an experiment where we've asked, okay, is knocking down H4K20 trimethyl and H4K20 dimethyl actually affecting chromatin state? And so we knocked down SUFR20H1 and H2, and we did our dual color fish, and we could see that if we knocked down these enzymes, that the chromatin got more open, and the distance between our two markers on chromosome 16 got bigger. So with this in mind, we also wanted to ask, if there's an H4K20 trimethyl CTCF access, and if it is reversible, we wanted to ask, what does it look like on a cell-by-cell basis? So among the proliferating cells, are there just some cells that are very extreme and some cells are not changing? Or do the cells basically all do the same thing? And how does this reverse when the cells are given the opportunity to re-enter the cell cycle? So we see here individual cells plotted for H4K20 trimethyl on the y-axis and CTCF on the x-axis. And you can see that the proliferating cells are here with low H4K20 trimethyl and high CTCF. When they become quiescent, they have much less CTCF, more H4K20 trimethyl. And when we restimulate them, we can see that they go to a place very similar to the original proliferating state. And in addition, each group seems to be quite homogeneous. So it's not that there are some cells doing something extreme and most of them aren't reacting. The cells seem to relatively homogeneously and reversibly shift along this H4K20 trimethyl CTCF somewhat antagonistic axis. So then we wanted to ask, well, is this just kind of a consequence of being in a proliferating state and a quiescent state, or is it possible that these molecules actually affect each other? So if we manipulate the levels of H4K20 trimethyl, for instance, will that affect the amount of CTCF that's there? So in this case, we asked, if we deplete this H4K20 trimethyl, does it affect this amount of CTCF that's bound to chromatin? So we used a molecule called A196, which is an inhibitor of the SUFR20H methyltransferases. And we found that if we gave H4K20 trimethyl, if we gave A196, the levels of H4K20 trimethyl decreased. And we asked what happens to CTCF levels. And you can see here that CTCF levels, shown in red, actually increased when we gave the cells A196, suggesting that A196, which inhibits H4K20 trimethyl, is sufficient to end up with more CTCF. So if you don't have as much H4K20 trimethyl, CTS will kind of fill the void. And we did this using immunoblotting as well, and we looked at the levels of CTCF, and you can see for chromatin-associated CTCF, we saw an increase when we treated with A196, and that's plotted here. Then we asked, does H4K20 trimethyl actually affect the cell morphology? So one of the key changes that we observe when cells become quiescent are changes in how they visibly appear. And that's shown here. So here we show that the nuclei of cells that have become quiescent from either serum starvation or contact inhibition get smaller. So here's nuclear area, and either contact inhibition or serum starvation, they're smaller. And then when they're restimulated, they get bigger again. And the nuclear aspect ratio changes. So here, when they're contact inhibited, the long axis to the short axis, it's greater when they're quiescent by either serum starvation or contact inhibition, and then they get kind of rounder, so a smaller nuclear aspect ratio when they're stimulated to proliferate. And we've also looked at nuclear volume, and we see that nuclear volume decreases as well when they're quiescent. In addition, we see that the nucleoli are smaller, so this is staining with fibrillarin, and when they're quiescent by either contact inhibition or serum starvation, which is shown here, we see less nucleoli, and when they're stimulated to proliferate, the nucleoli get larger again. So we found that inhibiting H4K20 trimethyl deposition actually did recapitulate many of these changes that we see with quiescence. So here you can see that the nuclear area gets smaller when we treat cells with serum starvation, but if we gave A196 to prevent H4K20 trimethyl deposition, the cells were actually not as small, the nuclei were not as small. We see the changes in the nuclear aspect ratio, so the cells normally get longer. If we gave A196, they didn't get as long, and the nuclear area was much larger, so you can see a lot more nucleolus if we gave serum starved cells A196. Then we asked, does H4K20 trimethyl actually affect quiescence gene expression? So I showed you that there are a lot of changes in gene expression that's shown here when cells become quiescent. We asked if we prevent H4K20 trimethyl deposition with A196, does that affect these gene expression changes? And as you can see, many of the genes that are induced when the cells become serum starved are inhibited if we prevent H4K20 trimethyl deposition with A196. And we've looked at these data with principal component analysis, and you can see here are the proliferating cells, here are the serum starved cells, and if we treat the serum starved cells with A196, then they shift and move more towards the proliferating state. Given these data, we also wanted to ask, well, these changes in CTCF differential binding, does that actually correlate with differentially expressed genes with quiescence? So I showed you at the beginning that there are a lot of genes that change in expression with quiescence, and we wanted to ask, all of these sites that we found CTCF is differentially bound, does that correlate with the specific locations where we see these differentially expressed genes? So you can actually see it here. We're showing the differentially expressed genes and the changes in CTCF binding. And you can see that actually visibly, it looks as though there's a correlation between them, between these yellow marks against the blue background and the red against the white. And we've looked at this kind of systematically and quantitatively, and we see that these TADs, these topologically associated domains that do have differentially bound CTCF sites do have statistically significantly more of the differentially expressed genes with quiescence. So showing that H4K20 trimethyl can affect CTCF, we then wanted to ask, well, is this kind of reversible? So if we manipulate CTCF, does that affect H4K20 trimethyl? So in order to test this, we first knocked down CTCF with siRNAs, and we found here a knockdown of CTCF versus a control siRNA. And we see that if we knock down CTCF, we end up with less CTCF. And we could see, yes, indeed, we have, when we do immunofluorescence for H4K20 trimethyl, we can see that we actually do have an increase in H4K20 trimethyl in response to reduced CTCF. So if there isn't as much CTCF, you'll get more H4K20 trimethyl in its place. And then we asked, does knocking down CTCF actually affect the cell morphology? And we saw that if we knock down CTCF, the cells kind of recapitulate what the serum start cells look like, so they get kind of longer and thinner. And the nuclei go from being rounder to being smaller, so here's the area, and more elliptical. So we end up with a bigger nuclear aspect ratio, the ratio of the long axis to the short axis. And we found, just like in quiescent cells, a reduction in the amount of nucleoli when we knock down CTCF. So then we wanted to ask, does CTCF knockdown affect gene expression? We saw H4K20 trimethyl affects gene expression. Does CTCF affect gene expression? And with the principal component analysis here, we saw something very similar. So here are the serum start cells, here are the proliferating cells. If we knock down CTCF, we can see that the proliferating cells shift more in the direction of the serum start cells. Then we ask, if we knock down CTCF, does it actually affect the cell cycle? And we found that knocking down CTCF was indeed sufficient to reduce the amount of proliferation that the cells were undergoing. Here you can see the amount of Ki67, a proliferation marker, is reduced when we knock down CTCF. And here you can see we added the siRNAs at about 24 hours at time 0, and you can see that the cells, it takes a little while to reduce the CTCF, and then the cells grow less. And here's our cell count per well. And we look at a really important marker for quiescence, P27, and we could see that when we treated the cells with the siRNA to CTCF, so it's not that they're just dying or stressed, actually they're inducing P27, which is a pretty specific marker for quiescence. So we can see that the cells are actually entering a quiescent state when we knock down CTCF. So then we wanted to ask, okay, knocking down CTCF, we get more H4K20 trimethyl, but what about overexpressing CTCF? Would this be sufficient to push the cells more to a more proliferative state? Would it reduce H4K20 trimethyl? Just overexpressing CTCF, would that be sufficient? And so in order to test this, we introduced either CTCF or a control vector into our cells. And you can see here we ended up with more CTCF. When we did this, we could see that actually CTCF levels were higher. And when we looked at H4K20 trimethyl, we could see that overexpressing CTCF seemed to be sufficient to kind of displace H4K20 trimethyl. And the levels ended up lower. So then we wanted to know, does CTCF overexpression actually affect the cell morphology? And as you can see from this kind of look at these cells, the nuclear area did increase and the nuclear aspect ratio decreased as the cells became rounder in response to CTCF overexpression. And we could see that actually we started to get more nucleoli and more abundant and brighter nucleoli, larger nucleoli when we overexpressed CTCF. And we wanted to ask, does CTCF overexpression affect gene expression? And we could see that when we compared with a control, when we overexpressed CTCF, the genes that were normally induced with serum starvation were not induced as strongly. And with the principal components, we could see here are the proliferating cells, here are the serum start cells. If we overexpress CTCF, then the serum start cells seem to be shifting more towards the proliferative state. And we asked, does CTCF overexpression affect the cell cycle? And so we took fibroblasts that were serum starved, so we removed their serum and we looked at Ki67 fraction. Here you can see even after 7 days of serum starvation, the cells that were overexpressing CTCF still had Ki67 positive cells, and we saw a statistically significant increase in the Ki67 fraction at 7 days in the cells where CTCF was being overexpressed. So overexpressing CTCF seemed to help the cells to resist entering a quiescent state. And here we see that P27, as I said, is a marker of quiescence. If we overexpress CTCF, we had a reduced level of P27. So we wanted to see if we could see specific examples in which the levels of CTCF and H4K20 trimethyl were actually regulating the expression of cell cycle genes. And so for cell cycle genes, in general, we expect high CTCF is going to lead to higher expression of these genes, And in our minds, we imagine that maybe without CTCF, we wouldn't be making these tags. With high CTCF, we would form the tags, and an enhancer would go and activate expression of a gene. So one example that we found is the gene for Ki67, which we said is a good proliferation marker. So here is the Ki67 gene, and we can see that flanking Ki67 are sites where there's either CTCF or H4K20 trimethyl shown here. And we can see that there's probably more CTCF when the cells are proliferating and maybe more H4K20 trimethyl when the cells are quiescent. So Ki67 expression is low when they're serum starved, it's high when they're proliferating. And we found that just overexpressing CTCF was sufficient to induce Ki67, whereas knocking down CTCF was sufficient to reduce the Ki67 levels, consistent with our model. And then we wanted to ask, is it possible that CTCF is affecting genes that are actually induced with quiescence? So can this go in the opposite direction? So in this case, we would expect, well, maybe with low levels of CTCF, maybe there's an enhancer that's outside the tag, so you would get more expression, or maybe there's a repressor for that gene within the tag. And so when there's high CTCF, you would have low expression because that repressor is reducing expression. And we did see a really nice example of this. So there's a gene called elastin, and elastin is a gene that encodes a protein, and the elastin is an extracellular matrix protein that helps to give our tissue elasticity. And the elastin gene is right here, and you can see it's flanked by 2 CTCF sites. And at those same sites, you can get H4K20 trimethyl, which is probably enriched in the quiescent cells, while the CTCF peaks seem to be higher in the proliferating cells. What we saw was that elastin expression is higher in serum-starved cells. CTCF overexpression slightly reduced it, and knocking down CTCF really increased the amount of elastin. So this was a case in which lower CTCF actually lead to higher elastin expression, which means that lower levels of CTCF and possibly higher levels of H4K20 trimethyl may be explaining some of the genes that are induced with quiescence, which was one of the questions that we really wanted to get at when we started this project. And then we also wanted to ask this change in CTCF and H4K20 trimethyl, this kind of mutual antagonism that we were seeing in cultured cells, is it also true in vivo? So is chromatin more open and our wounds and our CTCF more bound in proliferating fibroblasts in the context of wounds? So in this case, we've taken mice and introduced wounds into them and done electron microscopy on unwounded skin and wounded skin. And you can see in the unwounded skin, the nuclei had very condensed heterochromatin. When these fibroblasts were taken from the wounded area, the nuclei were much more open and kind of fluffy, less condensed, less electron dense. And so we quantified the electron dense area, and we could see that when the fibroblasts in this wounded area where they were starting to proliferate had less of the nuclei were electron dense. And we asked what happens to CTCF levels. So here we have mice where we've labeled the fibroblasts in green with a Cree-Lox model system using a promoter that is expressed from the collagen 1A2 promoter. And so anywhere that's expressing collagen 1A2 is going to be green, which is basically the fibroblasts in the skin. So the fibroblasts are green here. Here in the unwounded state, when we stain with CTCF, we really don't see a lot of staining of the fibroblasts. Here, this is distal skin, so not near the wound. We see relatively little CTCF staining of the fibroblasts. And here, right proximal to the wound for the fibroblasts that are proliferating, trying to heal the wound, we see higher levels of CTCF here in the fibroblasts, showing that when the fibroblasts are proliferating in vivo, we see higher levels of CTCF. And then we wanted to ask, if we reduce H4K20 trimethyl, does that promote proliferation in mouse models and in mice? So we took advantage of SUFR20H2 knockout mice, and we generated mouse embryonic fibroblasts from these mice. And you can see here with Western blot that when the fibroblasts were contact inhibited, we could see more H4K20 trimethyl. And if they were SUFR20H2 knockout and proliferating or contact inhibited states, we saw lower levels of H4K20 trimethyl. So when we grew these cells, we could see that actually the cell number was higher if they were SUFR20H2 knockout, so they seemed to proliferate more. And when we looked at the percent of cells in S phase, Here we see in the proliferating state, here we see that when they're contact inhibited. When they were contact inhibited, there were a lot fewer cells in S phase. And if they were knockout, then we had a higher fraction of cells in S phase, both in the proliferating and the contact inhibited states. We looked at the levels of cell cycle promoting cyclin, cyclin E1, cyclin D1, and cyclin D2. These tended to be higher when the cells were proliferating than when they were contact inhibited. And if the cells were SUFR20H2 knockout, we saw higher levels of each of these cyclins consistent with more proliferation. So then we asked, okay, what happens to the mice? If the fibroblasts are more proliferative, then do we actually get like more proliferative mice? So we looked at the embryos and we weighed them, and we could see that actually the embryos, this is day 14 embryos, were actually a little bit heavier. And we looked at the body weight of the mice that were born, and this is weeks 3 through 7 of their age. And we could see that actually for both males and females, the mice were heavier. So just to summarize what I've told you today, H4K20 trimethyl, which is a mark that had been really associated with heterochromatin, repetitive elements, and compact chromatin, and CTCF, we find can compete for chromatin boundary elements. We see that CTCF and H4K20 trimethyl play opposing roles in organizing chromatin, with more CTCF in proliferating cells, more H4K20 trimethyl in quiescent cells. We see that modulating H4K20 trimethyl, or CTCF, can reversibly shift cells between proliferative and quiescent states. And we see that mice with reduced H4K20 trimethyl are actually larger, which is consistent with extra proliferation. So our working model of proliferating and quiescent fibroblasts is that when the cells are proliferating, they have more CTCF, lower H4K20 trimethyl, they have more open chromatin, larger, rounder nuclei. When they're quiescent, they have low CTCF, more H4K20 trimethyl, they have more compact chromatin and smaller and elliptical nuclei. And we think that this helps us to answer some outstanding questions about proliferation and quiescence. So we wanted to know, why are quiescent fibroblasts spindle-shaped? And maybe this has to do with this process that we've investigated. We want to know what regulates the shape in cell and nuclear, the changes in cell and nuclear shape and chromatin compaction with quiescence. And we think that this model may help to address that. We wanted to know, how is it that different signals induce similar gene expression patterns? And we see that both contact inhibition and serum starvation lead to changes in the same pathway. And so this may help to explain why we see such similar gene expression patterns. And also, what regulates this quiescence gene expression program. So we know that this isn't everything. Only about 20% of quiescence regulated genes are in these topologically associating domains that behave in this way. So there's plenty of room for transcription factors, which we are also studying, and I hope I'll have a chance to tell you about in the future. But we think that this may help to explain at least some of the gene expression changes with quiescence. So huge thank you again to the organizers. Thank you to all of these institutions for funding our research. We're really grateful for their support. Huge thank you to all of the members of my laboratory. They were a fantastic team. They've all worked really hard on their own projects and on this particular project. Special thank you to Kaiser Tai, who's a graduate student who graduated recently, who really spearheaded this project. And a huge thank you to all the members of my laboratory for making science so much fun.
Thank you very much, Dr.
Thank you very much for the presentation, Dr. Collier. Now, Dr. Llanos will proceed to deliver the questions for the audience.
Great. I'm sorry, I can't hear you.
My apologies. There's a problem with the microphone. Thank you so much. Thank you so much for being here, for accepting our invitation. Now I will proceed to ask the questions from the audience.
Terrific. Thank you.
Thank you. So we have a question. What is the biggest technical challenge in inducing deep quiescence without leading the cell to irreversible senescence or apoptosis?
That's a great question. So So different cell types enter quiescence different ways. And for different cell types, there are different challenges. Some cell types that we've tried to make quiescent, they tend to apoptose, so they tend to die very easily. And so keeping them alive is a challenge. Some cell types, so we tried to grow some B cells, and keeping them alive was a challenge. I tried to do this in epithelial cells. For the epithelial cells, actually getting them quiescent was a challenge because the epithelial cells secrete their own mitogens, and so they activate themselves. And so even things that had worked for fibroblasts weren't working for the epithelial cells. Fibroblasts, the biggest challenge is they senesce on us. So for us, we can only use them for a limited amount of lifespan, especially because we don't use kind of transformed cells or genetically engineered cells. So we only have a certain amount of time before they're going to start to senesce. So that means that we need to be really rigorous about keeping track of what passage our cells are, using them for a limited amount of time, and keeping a close eye on them. So they'll senesce if they've had too much lifespan, but they'll also senesce if we just plate them too sparsely, or if they're unhappy in any way. So we have to really take good care of them and keep a very close eye on them and make sure that they're not senescing. And if they senesce, they have totally different properties and will turn on different genes. So that's a great question. And the answer is making sure that they don't senesce rather than quiesce.
Okay, thank you. I have another question. So, Dr. Kohler, in the field of health, the ability to control cellular quiescence is emerging as a tool, as a powerful tool, therapeutic tool with two sides, promoting it to protect and awakening cells to regenerate. From your experience, which do you believe is the most promising and viable strategy in the medium term? 1, inducing or strengthening quiescence in damaged cell populations, for example, in neurons after a stroke, or 2, awakening quiescent cells in a controlled and safe manner, such as progenitors, in tissues, or in oncological contexts, dormant cancer cells, to promote repair or make them vulnerable?
I think both. I think both are being actively pursued. I think they're both really important. I think they're both really exciting fields. We just had a conference of people who study quiescence, and it was so exciting to hear about all of the fantastic strategies being taken. And Alexis Barr actually laid out really clearly like her many-point plan for the ways in which she thinks that we can kind of use quiescence, in this case, in the case of cancer, but people are thinking about every which way. So for cancer dormancy, there's the question of, can we take proliferating cells and make them dormant? So we're trying to figure out what the best way is to do that. The other approach would be, can we take dormant cells and kill them? So we're trying to figure out, are there vulnerabilities of dormant cells, of dormant cancer cells that aren't vulnerabilities of quiescent normal cells that we can take advantage of to kill dormant cancer cells? And what you were suggesting, which is, can we take dormant cancer cells, stimulate them to proliferate, and then kill them with our chemotherapeutic agents, which mostly kill proliferating cells? So all three of these approaches we're actively pursuing. I think all of them are really promising. I'm not sure which one's going to come out the winner, but they're all things that I hope will allow us to better treat cancer and other diseases, improve regeneration in hopefully a pretty short period of time. I think it's a really promising, exciting field, and I think both of what you suggested is really promising and exciting.
Okay, great. I have another question, so let's go towards that. Dr. Keller, in your fibroblast model, the anti-correlation you observe between the genomic architecture organizer CTCF, height in proliferation, and the repressive chromatin marker H4K20Me3, height in quiescence, offers a fascinating mechanisms insight into cellular reprogramming. In fibroblasts, do you believe that the loss of CTCF and the gain of the H4K20Me3 at the specific LOPC primarily act to physically switch off promoters enhancers, of activation and proliferation programs, such as extracellular matrix genes and activated fibroblast markers, thereby reconfiguring the genome towards a quiescence state? Or does your data suggest a more direct role in the stabilized transcriptional silencing of these genes, where H4K20Me acts as an epigenetic lock?
Yeah, that's a great question. Thank you for the great question. So for any particular, so H4K20 trimethyl has been reported to go to promoters and to repress the expression of, for instance, E2F genes. That may be happening at some sites. And so for any particular gene, that could be the mechanism, and H4K20 trimethyl could be important at promoters or enhancers. Just we know that if we look like overall at the genes that have H4K20 trimethyl at their promoter in quiescent cells and ask, were they induced with quiescence or were they repressed with quiescence? 5% of them went up with quiescence and 5% of them went down with quiescence. So the overall, and yet if we give A196, we see this big shift in gene expression profile that looks more like the proliferating cells. So our thought is that we think that overall, this TAD formation and the boundary elements we think is more important than just sitting at promoters, just from the perspective that it seemed like it didn't really matter overall if we look at like what goes up and what goes down. So we think that the kind of boundary formation and forming those boundaries is very important for enhancer promoter interactions. And so we think that it's regulating a lot of enhancer promoter interactions through this mechanism and affecting quiescence through this mechanism, as I guess is that it's more important than just sitting at the promoters, just based on like the data that we have and just this correlation of if you have H4K20 trimethyl at your promoter, like did you go down in expression? The answer was like actually not really. And just to point out, Peter Adams has a nice paper on H4K20 trimethyl in senescent cells in genome biology, and he saw like when he looks at the promoters and asks like if H4K20 trimethyl is at the promoter, was that gene downregulated with senescent? And he saw more H4K20 trimethyl with senescence. Was that gene downregulated with senescence? He didn't see that effect either in the senescent cells.
Okay, let me check if we have something else. I guess that's all. Thank you so much for accepting our invitation again. It is our pleasure. And now I'm going to give the chance to Daniel to express our gratitude in behalf of the organizers. Thank you so much, Dr. Hillary. It was my pleasure and our pleasure. Great. Daniel, please.
We sincerely thank Dr. Koller for her excellent presentation, which I am confident has been valuable to everyone. Thank you for sharing your time, knowledge, and expertise in cell and molecular biology. We also thank all participants for joining us this morning. We invite all attendees to stay with us for the presentation of our next speaker.
Bye.
Buenos días. En esta oportunidad tenemos el agrado de presentar al magíster Lionel Germán Acosta, quien es director del laboratorio del Centro de Fertilidad Fertigestar y del Laboratorio de Biología Genética Molecular, además de ser jefe del Laboratorio de Citogenética Humana Genor y docente en la Universidad César Vallejo. El magíster Acosta nos presentará su ponencia sobre microRNA en espermatozoides y su correlación con la reproducción asistida. Magíster Acosta, sea bienvenido a este evento y agradecemos su participación y apoyo con nosotros. Magíster Acosta, le recordamos que cuenta con un máximo de 45 minutos para realizar su ponencia y luego se procederá a responder las preguntas durante un tiempo máximo de 10 minutos. Al público asistente en sala, les recordamos mantener sus micrófonos y cámaras apagadas para evitar interrupciones durante la exposición y las preguntas ustedes las formularán mediante el chat durante el transcurso de la presentación y serán absueltas al finalizar la ponencia. Además, les comunicamos que durante el transcurso de la exposición se les estará compartiendo por nuestras redes sociales el link del formulario para que registren su asistencia. Adelante, magíster Acosta, lo escuchamos.
Hola, ¿qué tal? ¿Cómo están todos? Salomón, gracias por la presentación. Igual agradecer a la doctora Fátima por la invitación. Vamos a tratar de colocar la pantalla. Por favor, un segundito. Y me confirman, por favor, si es que...
Se logra visualizar.
Un segundo. Ya, también se lo creo que así no es. Vamos a... Es pantalla completa, ¿no?
Sí.
Me confirman si pueden ver.
Sí, magíster, sí lo observamos.
Excelente. Entonces, nuevamente, agradecer a cada uno de ustedes por la invitación, para ser parte de este gran magno evento. A la doctora Fátima, a ti, Salomón, por la invitación. Vamos a conversar un tema muy relacionado actualmente, o muy visto en todo el mundo, que es la reproducción asistida, la fertilidad. Actualmente existe en el mundo 1 de cada 6 parejas en edad reproductiva que tiene este problema de fertilidad. Las causas realmente se conocen en muchos de ellos, pero se sabe que el 40% de los casos de infertilidad se debe a un factor masculino, otro 40% a un factor femenino y un 20% a ambos. Quiere decir que tanto el varón como la mujer llegan con problemas. Dentro de este mundo de la infertilidad, nosotros somos un centro de fertilidad que venimos atendiendo a muchos pacientes, y para que nosotros podamos hacer una técnica de selección espermática, ¿no?, hemos hecho muchas pruebas para determinar qué técnica sería la mejor opción a tomar para que nosotros tengamos los mejores resultados. A pesar que hacemos 1000 pruebas, hacemos muchas cosas, todavía nos queda la duda, a veces nos funciona, a veces no nos funcionan los resultados, y es por ello que nosotros, junto a Genética Center, que es una empresa hermana, hemos decidido hacer un poco más de investigación, es decir, llevar más allá de la evaluación del espermatozoide, que actualmente se realiza mediante un espermograma o un test de fermentación, evaluar a nivel molecular, a nivel de los microRNA, para determinar si realmente esto, como un biomarcador, nos puede ayudar a seleccionar la mejor técnica dependiendo la calidad espermática de los espermatozoides. ¿Sí? Entonces, actualmente ya se encuentra en los centros de fertilidad con la CIA. Vean este videito rápido, donde nosotros podemos ver detalle a detalle el desarrollo embrionario, determinar qué puede estar pasando con los embrioncitos, quiénes están cumpliendo una división a la hora indicada, quiénes están actuando más rápido, quiénes están dividiendo más lento, quiénes están formando a los mejores embriones, qué pasó con cada embrión. Entonces, todas estas preguntas nos llevan a entender también si solamente depende de la calidad ovocitaria o también está influenciado no solamente del ADN del espermatozoide, sino también de otros factores como los microRNA que pueden estar intuyendo a que tengamos a los mejores embriones. Perfecto. Entonces, ¿Qué es un microRNA realmente? Son pequeñas moléculas de ARN no codificantes, aproximadamente de 19 a 25 nucleótidos que van a regular la expresión génica de las células. ¿Y cómo actúan estos microRNA? Estos microRNA se van a unir al ARN mensajero, ¿okay?, para poder bloquear la producción de proteínas o poder degradar el ARN mensajero. ¿Qué quiere decir? Están regulando la expresión génica. Estos microRNA llegaron para poder controlar o están para poder controlar qué proteínas tienen que expresarse, qué ARN mensajero se tiene que degradar. Entonces, si ustedes pueden ver acá, una vez que tenemos el microRNA maduro, va a activar el RNA mensajero, va a inhibir la traducción o va a causar una deadenilación. Entonces, para entender un poco la biogénesis, antes de poder ingresar ya a los microRNA, ¿dónde se forman estos microRNA? El microRNA se forma en el núcleo, vean, y esto, gracias a una RNA polimerasa 2, van a formar unos primicroRNA. Estos primicroRNA luego van a sufrir nuevamente por esta enzima drocha con la proteína de GCR8, un corte y aproximadamente van a tener 85 nucleótidos y se va a llamar premicroRNA. Este premicroRNA va a salir, gracias a la esportina 5, va a salir hacia el citoplasma, y una vez que sale en el citoplasma, vean, va a actuar la enzima Lyser con el TRBP, que es la proteína de unión de ARN en respuesta a la transactivación, y lo que va a hacer es liberar este bucle, generando, ¿no?, un RNA doble. Luego viene la proteína argonauta, la cual va a separar esto en una hebra simple, y una de las hebras simples de este microRNA va a ser degradado, y la otra hebra de microRNA es la que va a formar el complejo silenciador inducido por ARN o el complejo RIS. Este complejo RIS es el que se va a unir a la región 3UTR del ARN mensajero. Una vez que se une a esta región, es lo que va a causar de que realmente se inhiba la traducción, cause la deadenación del RNA mensajero, haciendo que sea inestable y que esto se pueda activar, o el clivaje total del RNA. Entonces, ¿qué es lo que sabemos actualmente nosotros? Sabemos que todas las células de nuestro cuerpo tienen el mismo ADN, sin importar el tejido donde se encuentre. Es decir, todas nuestras células tienen la misma información genética. Pero ¿qué hace diferente que una célula del cerebro, las células óseas, las células sanguíneas tengan funciones diferentes? Hace de que tengamos genes activos diferentes en cada una de ellas. ¿Y quién hace o qué se hace responsable de que estos genes se estén activando y que cada una de las células tengan funciones diferentes? Se conoce actualmente que aproximadamente, en la gran cantidad de estos casos, son los microRNA los responsables que van a hacer posible de que estos genes se puedan activar. ¿Qué quiere decir? Hagamos un ejemplo y hagamos algo sencillo. Yo tengo la misma información genética en el cerebro como en el riñón. ¿Perfecto? ¿Qué quiere decir? Que en el cerebro los microRNA se están produciendo, ¿ok?, haciendo que solamente se puedan expresar los genes 1, 2, 5, 6, y en el riñón se están produciendo microRNA que se activen los genes 3, 4, 7. Entonces, al final todas las células tienen la misma información, pero gracias a estos microRNA hace posible de que se puedan expresar estos genes específicos. Dicho esto, entonces podemos saber, ¿no?, que estos microRNA que se están uniendo a los RNA mensajeros están inhibiendo la producción de una proteína. Y estos microRNA son muy importantes para que ocurra el desarrollo embrionario, la fisiología celular, e incluso actualmente están siendo muy estudiados como biomarcadores para enfermedades como en el cáncer. En humanos, la alteración de la función de estos microRNA se ha relacionado con diversas patologías, como el cáncer, enfermedades cardíacas, enfermedades endocrinas, enfermedades óseas e infertilidad. En realidad, nosotros vamos a basarnos al campo de la infertilidad. Cuando existe una sobreexpresión o hay una baja expresión de estos microRNA, podemos nosotros ya inducir como biomarcadores estos microRNA que algo está pasando y está llevando a la infertilidad, ¿no? Se sabe además que estos microRNA van a representar del 2 al 3% del genoma humano, que regulan la expresión aproximadamente del 60% de los genes, y además un solo microRNA puede regular más de 200 transcriptos. Como podemos ver acá, el microRNA 106B, 5P, ¿no?, puede estar regulando muchos transcriptos. Es un regulador importante. Y un RNA mensajero solo puede estar regulado por múltiples microRNA. Entonces, ya entendimos qué es microRNA. Gracias a los microRNA se están expresando muchos genes que están produciendo proteínas y que lleva a la diferenciación celular y que cada célula pueda producir genes diferentes y tengan funciones diferentes. Ahora, ¿cómo a nosotros en el campo de la reproducción asistida nos van a ayudar a entender un poco más qué es lo que está pasando y cómo nosotros podemos optimizar esos resultados para poder entender y llevar a término un desarrollo embrionario, tener el mejor blastocisto y por ahí tener, sin duda, un bebé sano en casa? Siempre se ha creído de que la única función del espermatozoide es llevar, transmitir el ADN, colocarlo dentro del ovocito y listo, con eso se acabó y aquí para adelante que sea el ovocito con el ADN del espermatozoide, no que se forme el embrión. Pero sabemos que más allá de eso, ¿okay? Más allá del ADN, el espermatozoide aporta más cosas. Entre ellos está poder introducir el factor de activación del ovocito, el poder colocar también proteínas, RNA mensajero, RNA secuencias no codificantes, incluidos estos microRNA. Entonces, quiere decir que hay más información de lo que uno puede tomar en cuenta. Y en realidad, hacer una extracción de estos microRNA es complicado. A nosotros, personalmente, cuando decidimos hacer este tipo de investigación, nos ha tomado mucho tiempo poder estandarizar esta técnica, poder recuperar estos RNA, poder recuperar estos microRNA que son importantes para su evaluación. Dentro de los estudios iniciales, primero quiero determinar, ¿hay realmente microRNA en el espermatozoide? Ya se había detectado por ahí unos cuantos, pero este es uno de los primeros trabajos de Albert Salas Huetos por allá en el año 2012, estamos hablando hace 13 años, donde determinó si que realmente habían más microRNA y que algunos microRNA o cuáles son los microRNA que podían encontrarse. Si ellos se dan cuenta y ustedes tienen un poco de conocimiento en el campo de la andrología, ¿no? Estos son los manuales antiguos donde clasificaba como astenosospermia, normosospermia, teratosospermia y normosospermia. Para que puedan entender, astenoterato significa que están lentos y tienen una movilidad, una morfología por debajo del 4%. En este caso, evaluaban la morfología por debajo del 14%, ¿sí? Hoy en día es difícil encontrar estos valores, pero bueno, este trabajo es importante, lo tomamos importante porque te dan como que las pautas, los inicios, cómo uno debería considerar el análisis para poder entender si estamos teniendo o no una expresión propia de microRNA. Entonces, ellos evalúan 4 pacientes, determinan la presencia y ellos, al evaluar los 4 microRNA, uno ya sabía que estaba descrito, que es el 23A, luego el 1744, se encontró el F7, pero el MIR1 no se encontró en espermatozoides. Y además se determinó que en algunos pacientes, como pueden ver, puede haber una sobreexpresión o puede haber una baja expresión. Entonces, esto te lleva a pensar que algo está pasando. Entonces, ¿por qué en algunos pacientes el mismo microRNA está sobreexpresado y en otros hay una baja expresión? Dentro de los trabajos, de ese trabajo, ¿no? La forma, nosotros lo tomamos en cuenta porque nos ayuda a entender cómo podemos tomar referencia de algo tan simple para poder nosotros tomar controles dentro del laboratorio. Entonces, ellos, ¿qué es lo que hacen? Van a usar a la protamina 1, ¿vean? Como un marcador para entender realmente, porque la técnica que utilizan para extraer ARN es simplemente trisol, no usaron kit, sino el trisol, y lo que hacen es, vamos a usar la protamina 1. Entonces, la protamina 1 cuando está en ADN tiene 422 pares de bases, pero cuando está en CNA tiene 331. Hay que entender que si vamos a hacer a nivel de los termocicladores, ¿ok? Solo podemos ingresar ADN, no ARN. Es decir, hay que extraer ARN, hay que convertirlos a CNA, y a partir de este CNA vamos nosotros a determinar la expresión, ¿no? En los real time de estos microRNA. Entonces, ellos hacen un control donde determinan, vean, que el ADN tiene 422, y luego todas sus pruebas corren con los mismos primer para poder determinar y demostrar que todos tienen 331 pares de bases que determina que no hay presencia alguna de ADN. Y de esta forma ellos confirman realmente de que lo que han extraído es ARN, lo que se han extraído es CNA, ¿sí? Se ha convertido en CNA y pueden evaluar la expresión. Luego hay un sinfín de trabajos que vienen evaluando a nivel de espermatozoide, a nivel de oocyte, a nivel de embrios, para que podamos nosotros determinar realmente la significancia y la importancia que puede tener estos microRNA como biomarcadores. Entonces, estos son todos los trabajos, ¿no?, de Attila Moskansky, en el cual ellos quieren determinar si realmente la evaluación de esta expresión de microRNA es la misma en plasma seminal, ojo, plasma seminal o en los espermatozoides. Y además determinar si es que esta expresión puede cambiar teniendo pacientes oligospermicos, que significa que tienen por debajo, ¿ya?, de 15 millones de espermatozoides, 16 millones de espermatozoides, o en pacientes astenospermicos que tienen por debajo de 30% de movilidad. Ellos al evalúan 11 microRNA, de los cuales determinan que 5 de ellos, vean, están sobreexpresados. Quiere decir, este es el paciente normal, como pueden ver acá están todos los normales, ¿sí? Todos los pacientes normospermicos están sobreexpresados, 5 de ellos, 5 de los 11, ¿no? Y que cuando tenemos un paciente ásteno o oligo, estos mismos microRNA se encuentran con valores muy bajos, que quiere decir que aparecen muy poco. Y esto nos lleva a entender, probablemente, de que no se está produciendo una buena cantidad de microRNA que está llevando a que se produzcan algunas proteínas, algunos mecanismos que llevan al proceso de infertilidad. Entonces, además, si se dan cuenta, tanto en el plasma seminal como en los espermatozoides, casi la concentración de estos microRNA siguen siendo iguales. Entonces dijimos 5 de los 11. Ahora, después determinaron que 3 de ellos estaban bajo regulados, que quiere decir que tienen que estar en una cantidad muy baja para que sean normospermicos, quiere decir pacientes que tengan una buena calidad seminal. Ahora entendemos de que no siempre los microRNA tienen que estar sobreexpresados. Pueden estar sobreexpresados algunos tienen que estar en una baja expresión para poder entender que la calidad espermática sea excelente. Más bien, en algunos casos, como en estos 3, como en el MIR 15B, 34B o 122, cuando había una sobreexpresión, ¿no?, estaba inhibiendo seguramente alguna proteína importante para que el espermatozoide sea considerado normal, tanto en movilidad como en morfología, como en cantidad. Entonces, y sin duda también habían microRNAs en el cual ni siquiera tienen una significancia. Es decir, la presencia pueden estar ahí, pero asisten en pacientes que tengan problemas o no está correlacionado con la infertilidad. Hay trabajos también que evalúan estos microRNA con relación a la fragmentación de ADN. Hoy en día se habla de fragmentación de ADN. No conozco ningún centro de fertilidad en el mundo actualmente que no esté haciendo esta prueba. Entonces, mucho va a depender. Algunos le van a pedir de rutina, otros le van a pedir después de hacer algún procedimiento o esperando que tener los resultados previos. Entonces, vamos a determinar si realmente esta fragmentación de ADN está o no involucrada con los microRNA. Pero incluso actualmente se discute mucho el porcentaje de fragmentación, ¿no?, a partir de cuánto es considerado realmente fragmentación de ADN. En este trabajo lo consideran fragmentación de ADN bajo, por debajo de 15, y fragmentación de ADN por debajo del 30, por encima del 30%. Entonces, ellos van a evaluar más microRNA para determinar si es que estos pacientitos que tienen un daño elevado de fragmentación de ADN realmente están correlacionados con estos microRNA. Lo hacen en 2 tandas. Primero evalúan los 18 marcadores con 24 pacientes. De estos 24 pacientes, ellos encuentran que 3 de ellos tienen una diferencia significativa en cuanto a la expresión de los microRNA, y luego ya juntan todos los pacientes, así con los 70 y luego con los 94 pacientes, para determinar realmente cuál de estos microRNA es el que tiene o no una correlación. Entonces, el título es, ¿no?, MIR 424 y su homólogo en animales, el 322. Entonces, vean acá, ¿no? El trabajo, analizan los 18 microRNA, determinan que el MIR 15B, que el MIR 424, el que estamos haciendo el estudio, y el 29C, realmente son los que tienen una diferencia significativa entre pacientes que tienen fragmentación por debajo de 15% y fragmentación de ADN por encima del 30%. Luego los juntan y determinan que en los 70 pacientes restantes, ¿no?, ya el MIR 15B pierde esa significancia y solo se quedan con el 424 y el 29C. Como pueden ver, vean, en pacientes, ¿no?, con alta fragmentación y pacientes que tienen baja. Entonces, quiere decir que en pacientes que tienen alta fragmentación, estos microRNA tienen una baja expresión. ¿Qué quiere decir? Que no se está produciendo adecuadamente, que probablemente se está produciendo alguna proteína que está siendo causante del daño a nivel de ADN. Ahora, ellos no se quedan ahí, ellos quieren hacer pruebas en ratones, quieren determinar si realmente este 1424 o el 29C, en realidad podemos nosotros traspasar a estos ratoncitos para determinar si realmente tiene o no un valor de significancia a nivel de ADN. Entonces, ellos hacen unos lentivirus en los cuales tratan de inhibir esta expresión y determinan, vean, cuando la expresión de 424 y en humanos o 322 en ratones, cuando se inhibe, la fragmentación del ADN empieza a aumentar. Y de esta forma determinan ellos que realmente este microRNA está muy representativo para poder decirte que la baja expresión de estos lleva a daño a nivel de ADN. Más allá, ellos quieren también determinar si es que a nivel de la histología, a nivel de FISH, hacen un marcador, el gamma H2AX, que es un marcador de doble daño a nivel de ADN. Si es que inhibimos el 322, van a haber células con más daño de doble hebra. Entonces, cuando colocan PBS, vean, normalmente no se observa casi nada, pero cuando empiezan a colocar o inhiben, ¿no?, este microRNA, el 322 o 424, se observan una gran cantidad de células con daño a nivel de ADN. Y con esto ellos pueden concluir que la inhibición de este microRNA realmente lleva a que exista daño a nivel de ADN espermático. Hemos estado viendo a nivel de espermatozoides. Ahora vamos a ver ya con su correlación espermatozoide y embriones. ¿Qué es lo que sucede realmente? En este trabajo de Lunkul, el espermatozoide van a evaluar al MIR34B y MIR34C. Son dos microRNA importantes también presentes en el espermatozoide y quieren ver que después de ser estos seleccionados e inyectados a los ovocitos, tienen o no alguna correlación con el desarrollo embrionario en día 3 y si podemos a partir de ellos determinar un punto de corte a partir del delta CQ para que podamos nosotros decir, mira, pacientes que inicialmente llegaron a un centro de fertilidad, se pueden analizar, se hace una extracción de ARN, se evalúa la expresión y podemos desde antes ya determinar si realmente este paciente tiene una buena probabilidad de embarazo o no. Entonces, es muy bueno porque te va a decir, mira, ya podemos usarlo como biomarcador. No solamente vamos a enfocarnos en cómo está la calidad espermática, sino evalúa la expresión de estos microRNA y según el punto de corte nos dirá, perfecto, este paciente tiene buena chance de formar buenos embriones. Entonces se determinó que el MIR34C, ¿no?, cuando tenía una mayor expresión, ¿no?, había una mayor cantidad de embriones que podían llegar a día 3. Ojo, este es un trabajo antiguo, día 3. Hoy en día los centros de fertilidad ya vamos a blastocisto, ¿no?, día 3. Y además ellos determinan de que el MIR34C tenía una mayor correlación bajo la curva, ¿no?, de 0.75. Y además ellos vean, a pesar de que expresaba una buena cantidad de MIR34C, había algunos que embarazaban y algunos que no. Y bajo los estudios ellos determinaron de que para aquellos que habían logrado embarazar, si tenía un punto de corte de 6.82 a nivel de expresión, entonces a nivel del delta CT, podíamos nosotros decir de que tenía buena probabilidad de formar buenos embriones y poder lograr un embarazo. Entonces ya empezaron a usarse los microRNA como biomarcadores, como pruebas iniciales, para entender que si el paciente tenía valores muy bajos sobre este punto de corte de 6.82, ¿no? Entonces podíamos hacer un tratamiento adecuado para mejorar la calidad espermática, hacer pruebas iniciales y ver si realmente este microRNA es importante o no. Entonces, este microRNA que estaba en espermatozoides ya se puede entender que puede estar también formando parte del ovocito o no. Entonces, para eso empiezan nuevos estudios y dicen, vamos a ver si es que realmente a nivel de ovocitos hay este microRNA. Y en este estudio de Zeng-Chi, ellos empiezan a tomar ovocitos que no fueron fecundados, células granulosa, y embriones que fueron triploides, embriones que se arrestaron, y determinaron que este 34C no se encontraba ni en ovocitos, no se encontraba ni en célula granulosa, ¿sí? Ni en huevos que no habían sido fertilizados, pero sí a partir de aquellos que ya habían sido fertilizados, entendiéndose entonces que este Mi34C es, ¿no? este microRNA es exclusivo de espermatozoides. Entonces, que la presencia de espermatozoides, ¿no? Y que lo va a llevar, esta expresión lo va a llevar al desarrollo embrionario, podría tener una significancia para tener a los mejores embriones, para tener a embriones de mejor calidad e incluso tener mejores embriones para la tasa de implantación. Y ellos empiezan a estudiar estos microRNA y también determinan un nuevo punto de corte. Dicen que quienes lograron mejores tasas de blastocisto, las tasas de embarazo, ¿no? La tasa de implantación era cuando el valor era mayor a 1.9. Entonces, nuevamente, estos microRNA tienden a tener una gran importancia para poder nosotros explicar realmente a los pacientitos. Es decir, ya no solamente evaluar al paciente con un espermograma, con una fragmentación de ADN, sino antes de acudir a un centro, ¿no? Antes de hacer una técnica de reproducción asistida, poder medir a nivel de expresión de estos microRNA y decir, mira, me salió tanto el valor, las probabilidades van a ser bajas, esperemos un poquito más, hagamos un tratamiento adecuado al semen, veamos si mejora la calidad seminal, veamos si mejora la expresión de estos microRNA y con estos valores como que podamos optimizar realmente la tasa de implantación o de bebé nacido en casa. Luego se ha evaluado, ¿no? Se ha tratado de ver también que no solamente sea la evaluación de un solo microRNA, sino empezar a evaluar ya microRNA en parejas. Hay muchos trabajos que determinan que la causa de infertilidad no solamente depende de un solo microRNA, sino que están involucrados en pareja, o de 2 o de 3, ¿no? Para poder saber realmente la expresión. Entonces, en este trabajo se evaluaron 106 pacientes infértiles, se hizo la fragmentación mediante la técnica de túnel, y se evaluó el 34C nuevamente, que está muy importante a nivel de espermatozoides, y en este caso el 449B5P. Y esta vez lo hicieron por digital PCR. Entonces, ellos determinaron que la expresión de estos microRNA mucho va a depender. Cuando encontraban una alta expresión del 34C, vean, pero a la par encontraban que el 449B tenía una baja expresión en el espermatozoide, el desarrollo embrionario era completo y podíamos obtener buenos blastocistos. Entonces, tenía que ocurrir esto, que el 34C esté bien y que el 449, ojo, ¿sí? Esté en bajas expresiones. Pero cuando ocurría al revés, que el 34C estaba en baja expresión y el 449 se expresaba mucho, la posibilidad de que llegue a un blastocisto, o sea, la última etapa del desarrollo embrionario, era casi nula. Entonces, entendieron que el 34C nuevamente es un marcador importante, que su alta expresión te ayuda a entender, a tener buenos embriones, y que el 449B realmente es un microRNA en contra del desarrollo embrionario, ¿no? Tanto así que cuando había mayor expresión del 1034C, en comparación con el 449, la tasa o posibilidad de que llegue a blastocisto era 14 veces más en comparación al resto. En este trabajo de Wahoo, lo que se hace y se determina realmente es poder determinar no solamente ya unos cuantos microRNA, sino una gran cantidad de microRNA y determinar en aquellos pacientes que tuvieron buenos resultados. Y esto también, primero, a ver si es que tienen resultados en la tasa de fertilización, en embrión efectivo y en embrión de alta calidad. Entonces, ellos empiezan a comparar y dicen, perfecto, vamos a separar grupos que tienen una buena tasa de fertilización, una buena cantidad de embriones, ¿okay?, que sean efectivos y una alta calidad de embriones. Al comparar estos grupos, encuentran una gran cantidad de microRNA, pero entre todos ellos, si se dan cuenta, ven que uno se repite, que siempre va a estar presente cuando tengamos buenos resultados de fertilidad, el 1191-5P. Si tenemos un embrión efectivo, tenemos nuevamente al 1191, y si tenemos un embrión de alta calidad, también está el 1191-5P. De esta forma, ellos dicen, perfecto, si se repite siempre, es porque este microRNA es importante para que pueda ocurrir todo este proceso. Y este proceso solamente está, o este microRNA está también en espermatozoides. Ojo, el hecho de que tengamos una buena alta expresión de este microRNA no quiere decir de que el que no lo tenga no va a llegar a blasto, no, porque son más microRNA que actúan, pero sí podemos nosotros deducir de que la alta expresión de esto nos lleva a pensar que este caso va para bien, ¿no? Entonces empiezan a evaluar, a determinar y evalúan realmente si está correlacionado también con la morfología, con la concentración espermática. Entonces llevan a entender de que realmente la expresión de este microRNA 191-5P está correlacionado realmente con buenos resultados mediante una fecundación in vitro. Lo curioso acá, que también evalúan el 34C, y ellos encontraron que el 34C, ¿no?, que sí había sido estudiado con buenos resultados, que tenían puntos de corte muy buenos, no había correlación con ninguno de los grupos. No había una buena correlación con la tasa de fertilización, no había una buena correlación con la tasa de embrión efectivo, con el desarrollo embrionario o la alta calidad. Imagínense. Entonces, esto también te puede llevar a pensar de que va a depender mucho de la población que vas a trabajar. No podemos comparar una población de Europa con una de Asia ni con una de América. Entonces, es importante que cada una de las poblaciones podamos determinar qué microRNA puede estar involucrado en tener a los mejores embriones y al mejor desarrollo embrionario. También se empezó a estudiar, no solamente ya a nivel de la célula, sino dijeron, bueno, estudiemos ahora el medio donde estaban involucrados los embriones. Si realmente existe posibilidad de que aparezca este 1191-5P, que esté sobreregulado en los medios de los embriones de mejor calidad. Entonces empiezan a comparar, a tomar los embrioncitos, cultivos individual, y una vez que determinaron qué embriones fueron de alta calidad, tomaron estas gotitas, analizaron los microRNA y determinaron realmente si es que en embriones que habían logrado implantar había una mayor expresión. Ellos analizaron el 21.3P, 24.15P, el 191 y el 322. Y vieron de que el 191 nuevamente, ¿no?, en los medios donde habían estado esos embrioncitos que habían logrado una alta implantación, se encontraban en mayor cantidad. O sea, la expresión de estos embrioncitos y de aquellos embrioncitos que no lograron implantar, este marcador 191.5P, ¿no?, se encontraba en baja expresión. Entonces, nuevamente empiezan a radicar otro marcador, el 34C que comentamos inicialmente y el 191 como puntos importantes. Hay muchos más marcadores, sin duda, hay muchos más microRNA que son importantes para que podamos nosotros determinar si realmente tiene o no un proceso efectivo o no en el desarrollo del espermatozoide o la importancia del espermatozoide. Pero estos dos han determinado estar presentes en espermatozoides y que tienen una significancia en el desarrollo embrionario. Y es por eso que nosotros en Gestar, en Fertigestar, junto con Genética Center de empresa hermana, decidimos hacer un estudio sobre ello. Dijimos, bueno, perfecto. Uno de los grandes problemas que tenemos nosotros en laboratorio es que existen varios modelos de técnicas de selección espermática. Entonces, ¿qué técnica es la mejor? Y si esa técnica realmente es válida para poder ser utilizado en todas las muestras. Es decir, yo le voy a usar un gradiente de densidad a todas las muestras, así sea normo, ásteno, terato, oligo. ¿Debería ser eso? ¿O debería cambiar a un swing A, por ejemplo, en todas, o también igual por separado, o las columnas de anisina? ¿Sí? Y en un momento nos gustaría también trabajar con Sino para determinar, ya que es una técnica que está de moda, ¿no?, los microfluidos que están ayudando a obtener los mejores resultados. ¿Qué es lo que sucede? Que nosotros inicialmente hicimos un trabajo propio, interno, y determinamos que si usamos gradientes, usamos columnas, usamos swing up, ¿no?, y lo queremos determinar cuál es mejor mediante el test de fragmentación de ADN, mediante la técnica de la dispersión de la cromatina, no encontramos resultados. Nos decían que en realidad por ahí no son significativos los valores y que yo podría utilizar cualquiera. Pero en realidad nos quedó mucho más duda. Dijimos, bueno, ¿será cierto eso? Y es por ello que decidimos, ¿por qué no estudiamos más allá? Es decir, analizamos más a fondo al espermatozoide. Empezamos a tener una muestra, empecemos a seleccionar la muestra por gradiente y SYNAP, evaluemos a la muestra inicial con estos microRNA, luego a la muestra seleccionada con estos microRNA y veamos si realmente el doble delta C realmente te dice cuál técnica es mejor dependiendo de la calidad espermática. Entonces, ese es el trabajo. ¿Y nosotros qué marcadores estamos usando? Hemos usado estos marcadores que hemos venido conversando, el microRNA 191.5P y 34C. Y no solamente evaluar con el desarrollo embrionario, sino también ver con el número total de espermatozoides, con la movilidad espermática, con la morfología estricta llamada MAI, y también con el índice de fragmentación usando la técnica de la dispersión de la cromatina. Entonces, ¿cuál es el objetivo? Por ejemplo, el objetivo final del trabajo que nosotros queremos tener en cuenta es que cuando llegue un pacientito y lo diga, mira, el paciente es oligoastenoteratospermico, perfecto. Ya nosotros hemos determinado que si el paciente es oligoastenoteratospermico, entonces la mejor opción es hacer swing up, un ejemplo, ¿no? Porque conociendo swing up, ya hemos determinado en nuestro estudio que vamos a obtener una mayor expresión de estos microRNA que son importantes para el desarrollo embrionario. Pero no, el paciente, hagamos el ejemplo, es normosopérmico, entonces sale un gradiente, ¿no? Una técnica de selección gradientes porque hemos visto que realmente el gradiente es mucho mejor, se obtiene mejor expresión de estos microRNA. O quién sabe, probablemente me digan, no, mira, al final úsale a todo gradiente porque el gradiente es mejor que todas las técnicas, o el swing up es mejor que todas las técnicas, o las columnas. Hagamos lo que hagamos, siempre se obtuvieron mejores resultados. Entonces, esa es la intención, de poder determinar qué técnica es la mejor, dependiendo el tipo de muestra, la calidad seminal, ¿sí?, para que nosotros podamos obtener mejores resultados en el desarrollo embrionario y tener las mejores tasas. Y sin duda, como les mencioné, hay una gran cantidad de estos microRNA que están relacionados en todo el proceso, en toda la diferenciación de las células germinales, la iniciación de la meiosis, en la misma espermiogénesis, que son responsables para que el espermatozoide se pueda producir. No solamente en eso, sino también en la parte de la ovogénesis también es importante, ¿no? Vean esta imagen donde hay una gran cantidad de genes estudiados, hay microRNA estudiados, hay una gran cantidad de papers que han salido y publicados, ¿no?, en la ovogénesis, incluso hasta en la implantación embrionaria. Uno de los trabajos que tenemos posa esto es determinar a nivel endometrial cuáles son los microRNA que según su expresión nos pueda llevar a entender, ¿sí?, no solamente haciendo la progesterona o haciendo la evaluación del útero, sino también a nivel molecular, decirnos, oye, perfecto, mira, la expresión de este microRNA está excelente, así que a partir de este corte, las probabilidades de embarazo empiezan a aumentar, ¿no? Entonces, ese es el objetivo que estamos teniendo nosotros actualmente en nuestro laboratorio, de poder trabajar con estos microRNA que están regulando muchos genes y que nos puede servir como biomarcadores para poder lograr el objetivo en nuestro laboratorio a diario. Entonces, como para terminar, ¿qué nos llevamos a casa? Lo primero que ya entendemos que existen diferencias a nivel de expresión de microRNA entre pacientes normospermicos y los que presentan algún parámetro alterado. Ya lo sabemos que existe eso, pero realmente a veces cuando llegan nuestros pacientitos, ya tienen 39, 40, 41 años, y mandarlos a hacer un tratamiento a veces es complicado, porque a mayor edad de la mujer, en realidad, empieza a haber menos posibilidad de lograr un brazo. Entonces, sabiendo que existe eso, por eso la decisión de poder tomar una técnica de selección espermática que minimice ese tiempo y que nosotros, haciendo esta selección, podamos entender que podemos obtener espermatozoides selectos con la expresión de microRNA deseados para formar embriones excelentes. Los niveles de expresión de algunos microRNA en espermatozoides están correlacionados con la calidad embrionaria, sin duda. Lo acabamos de ver que el 34C, el 191 y otros, que no he mencionado porque no lo estoy tomando en cuenta dentro del estudio, hemos visto que estos microRNA son importantes. Incluso cuando los pacientes se pueden dar el lujo de poder seguir un tratamiento porque son pacientes jóvenes que no logran embarazar, podemos hacer eso. Viene el paciente, determinamos la expresión, sabemos ya puntos de corte y el paciente puede tranquilamente recibir un tratamiento adecuado de 3, 6 meses, hacer nuevos controles y determinar realmente si es que aumentó esta expresión de microRNA y así nosotros poder entender que el resultado puede ir bien. Ojo, el hecho de que tengamos una mejor expresión no es que sea una regla de 100, no vas a tener resultados, digamos, ya te mejoré, vas a lograr el embarazo, vas a tener... No, hay muchos más factores, pero que es un biomarcador que te está ayudando a entender un poco más cómo va a desarrollarse ese embrioncito. Las investigaciones han determinado que los microRNA pueden utilizarse como biomarcadores para medir el grado de fertilidad del varón y poder realizar tratamientos previos antes de realizar una técnica de reproducción asistida. Y siempre invitar a los centros a realizar investigación en el campo de genética, biología molecular e implementar en su centro de fertilidad como un nuevo laboratorio de investigación. Actualmente ya todos los centros en el mundo, y nos incluimos acá en Perú, muchos de los centros ya no solamente hacemos la parte de diagnóstico, sino también están implementando los laboratorios de investigación, están trabajando en diferentes ramas, así como nosotros trabajamos con los microRNA, hay otros laboratorios que trabajan con maduración in vitro, hay otros laboratorios que tienen otro punto de vista, y todos empezamos a trabajar con la intención de poder mejorar. Y el único objetivo que tenemos los centros de fertilidad es que nuestros pacientitos puedan lograr ese embarazo. Nosotros a diario vemos muchos pacientes, vemos muchos tipos de muestras, tratamos de hacer estudios básicos incluso a nivel de ADN espermático, a nivel de calidad espermática, pero estamos sumando ya la parte de biología molecular y genética que sin duda nos va a ayudar a entender la mejoría de estos y los resultados al final. Muchísimas gracias.
Muchas gracias por su presentación, Magister Acosta. A continuación procederemos a transmitirle las inquietudes del público asistente. Tenemos varias preguntas. La primera es, ¿cómo puede afectar un perfil alterado en microRNA en espermatozoides a la calidad seminal?
Como mencionamos, primero hay que evaluar un paciente endospermico para generar realmente y saber cuál es el nivel de expresión. Los microRNA, como te decía, lo que van a hacer es inhibir la producción de algunas proteínas importantes. Entonces, si es que este microRNA, vamos a un ejemplo, tiene que estar sobreexpresado, que quiere decir, tiene que estar silenciando alguna proteína, y al final esta proteína está involucrada, por ejemplo, en que funcione, no sé, vaya mal para el espermatozoide, entonces no va a ir mal. Entonces, la intención de nosotros, de estos perfiles diferentes de poder generar, es poder entender cuáles son los microRNA responsables realmente de mantener una calidad espermática normospermática, y si es que esta variabilidad puede llevar a que sea astenoterato, oligo, ¿no? O cualquier cosa que pueda presentar la calidad espermática.
Muy bien, tenemos otra pregunta, la cual es, ¿qué obstáculos existen para implementar el análisis rutinario de microRNA en espermáticos en laboratorios de fertilidad en Perú, teniendo en cuenta recursos, costo y acceso a tecnología adecuada?
Exacto, sí, ese es uno de los detalles, ¿no? Implementar un laboratorio de investigación no es nada barato, ¿no? A nosotros particularmente nos ha llevado muchos años en tomar esa decisión. Estamos hablando de cifras por encima de los 6 números, ¿no? De poder implementar. Sabemos cuánto cuesta un termociclador, sabemos cómo cumplir con todas las normas, pero es difícil, es uno de los obstáculos muy difícil. Nuestra intención dentro de esto no es poder hacer como pruebas de rutina, más que eso, sino nosotros desde el laboratorio, una vez hecha la investigación, poder determinar y decir, como te mencionaba, mira, tengo un paciente que es oligoásteno, ¿no? Pero yo he determinado con investigación que este oligoásteno es mejor hacerle columna de nexina, porque haciéndole columna de nexina voy a tener mayor expresión, ya está determinado esto. O es un paciente normo, ¿no? Entonces, no es que lo vaya a mandar siempre a hacer estas pruebas, sino que ya me sirve como una base de datos para que yo pueda tomar las mejores opciones, porque a veces sucede que en laboratorio, ok, perfecto, ¿qué técnica le hacemos? Y trabajamos con todas las técnicas. Y a veces decimos, bueno, dale un gradiente, dale un swing-up, ¿no? Y con eso basta, pero al final vamos a los resultados y no eran los que esperábamos, ¿no? Entonces, ya con esto, teniendo un biomarcador más, que son los estudios de los microRNA, podemos decir ya con certeza, porque tenemos un estudio, este paciente que es terato, hale sí o sí un swing-up, porque voy a obtener espermatozoides que tengan mayor expresión de microRNA específicos para el desarrollo embrionario, ¿no?
Muy bien. Tenemos otra pregunta del público, la que nos dice, ¿en qué consiste el tratamiento para mejorar la expresión de los microRNA?
Los tratamientos van a ser múltiples. Siempre que un pacientito llega, ¿no? Primero tiene que pasar una consulta andrológica, ¿no? Porque tiene que ser evaluado, ser determinado. Ya el andrólogo tiene que determinar realmente, dependiendo de estos resultados, si le da un antioxidante o algún tipo de tratamiento, ¿no? Secundario para que pueda ayudar a metabolizar un producto o una proteína que sea necesaria. O sea, no es que vamos a ir y a todos le vamos a dar antioxidantes, no. Esto tiene que pasar por un médico especialista y determinar también, porque esto está asociado también a la calidad espermática. Imagínate que le dimos al andrólogo, le informamos, mira, este pacientito, además de ser astenospermico, presenta una gran cantidad de espermatozoides que sean amorfos o piriformes o elongados. Entonces, el médico entenderá que algo está fallando en la espermatogénesis y los andrólogos médicos, ya no descarto yo mi lugar ahí porque no soy médico, sabrán dar los medicamentos adecuados para mejorar y de esta forma mejorar la calidad espermática y que se pueda asociar a esta expresión de microRNA, ¿no?
Otra pregunta, Magister. ¿La expresión de microRNA tiene que ver con los bebés con TEA, que es el trastorno del espectro autista?
No, no tiene nada que ver. Es decir, hay trabajos, incluso justo revisaba unos trabajos recién, que dependiendo también, bueno, en el trabajo mencionaban que dependiendo la persona, la expresión de estos microRNA, podía llevar a depresión a los hijos. Imagínate, podía transmitir la depresión a los hijos. Mucho se ha estudiado si es que realmente el efecto, no solo del microRNA, sino de la técnica de reproducción asistida, tiene o no algo que ver con alguna enfermedad futura o los bebés que puedan tener algún problema. La ciencia ha determinado que no, que las técnicas de reproducción asistida llegaron para ayudar, que más puede estar relacionado porque son pacientitos que acuden a un centro de fertilidad, que tienen ya un problema desde antes, ¿no? Recuerden que los pacientitos que llegan es porque no pueden orar el embarazo de forma natural. Entonces, más que una técnica, más que determinar los niveles de expresión, y no tengo mucha experiencia en esto de DHA con microRNA, pero quién sabe, quizás al final, sí, ¿no? Los niveles de estos microRNA ya estén estudiándose a ese nivel de que no solamente en espermatozoides, puede ser algún microRNA en ovocitos, o sea, no podemos culpar todo el espermatozoide. Recordar que el 50% de la información genética viene del espermatozoide y 50% del ovocito, ¿no? Y hay que ver la expresión propia en el embrión, ¿no?
Muy bien, Magister Acosta. En su experiencia, ¿cuáles son las mayores dificultades técnicas para el aislamiento, cuantificación y validación de microRNA a partir de muestras de espermatozoides humanos?
Mira, el espermatozoide es la célula más hermosa que conozco. Es una célula muy, muy hermosa y a la vez muy difícil de estudiar. Hay que entender que, a diferencia de una célula normal o somática, este espermatozoide tiene una compactación 6 veces más, y esto porque hace un cambio de histonas por proteínas de transición y luego por protaminas que hacen 6 veces más que estén compactadas, y hace más difícil, ¿no?, la ruptura o la lisis y la extracción de estos ARN y estos microRNA. Es complicadísimo. Yo he probado particularmente varias técnicas, he usado el trisol solo, diferentes kits, y al final hemos tenido que generar una propia solución de lisis, digamos así, básica, inicial, previa, y calentar el grisol, es todo un sistema, no es tan fácil, las condiciones de tu laboratorio, mantener, sabiendo que el ARN es delicado, porque se degrada rápidamente, no usar las adeneasas adecuadas para que el ARN se mantenga, hacer los controles, es solamente la estandarización de extraer ARN a nosotros particularmente, y quizás alguien tenga más experiencia, genial, pero a nosotros nos ha llevado más de 1 año y medio prácticamente en estandarizarlo, para luego poder recién ver la expresión. Y ahora viene nuevamente, ¿qué kit vas a utilizar? Hay un mercado, hay una gran cantidad. Cuando las investigaciones son dadas por, digamos así, coloquialmente, de tu bolsillo, cuesta. Cuando son hechos por una entidad, genial, gasta lo que quieras, ¿no? La entidad te va a dar $1 millón y gástalo, ¿no? Pero cuando sale del bolsillo del investigador, como es en nuestro caso, entonces tenemos que tener mucha cautela, y antes de intentar nuevamente hacer algo, tenemos nosotros que entender, leer, investigar y luego probar. Entonces, esa es una de las limitaciones, ¿no? Que actualmente la investigación en el Perú, si no es por una universidad, ¿no? Y entidades privadas, es complicado, es complicadísimo. Pero a pesar de ello, sabemos la importancia que radica las investigaciones y estamos dispuestos a hacerlas, ¿no?
Muy bien, magíster. Última pregunta. ¿Existen perfiles específicos de microRNA espermático que usted y su equipo hayan identificado como correlacionados consistentemente con parámetros de calidad seminal alterados, como fragmentación de ADN, alta, baja movilidad o morfología?
Nosotros, como digo, no estamos analizando, solo analizamos 2 microRNA. Solamente estamos trabajando con el 34C y 191. Lo que sabemos de los perfiles de expresión es por los papers ya publicados. Pero no trabajamos con todos, no, es una barbaridad. O sea, ya trabajar con un solo microRNA cuesta miles de dólares, trabajar con 10, nos empeñamos todos 20 años de trabajo, ¿no? Nunca acabamos. Entonces, la investigación en esto a nivel de microRNA hoy en día está muy sobrevalorado, está muy costoso y es difícil de hacerlo. Entonces, por eso es que hemos tenido una vez que nos sentamos, conversamos y vimos y analizamos, determinamos solamente analizar 12. Te cuento que al inicio quería trabajar con 5, que tenía 5 publicados, pero te doy una cifra nada más, que trabajar con 5 sale algo de $120,000, $150,000, ¿no? Entonces es caro. Entonces decidimos bajarnos a 2 de ellos, ya es de decisión propia científica, y dijimos, vamos a analizarlo y lo estamos trabajando. Todavía no puedo dar los resultados porque es parte de una investigación, pero vamos bien, ¿no? Vamos por buen camino.
Muchas gracias, magíster Acosta. Agradecemos al magíster Acosta por su muy buena presentación, que estoy seguro ha sido provechoso para todos. Gracias por darnos su tiempo y transmitirnos sus conocimientos y experiencias en el área de la biología celular y molecular. Agradecemos también a los participantes de esta mañana por su asistencia. Invitamos a todos los presentes a continuar acompañándonos con la presentación de nuestro siguiente expositor. Muchas gracias, magíster Acosta.
No, a ustedes, muchísimas gracias. Encantado.
and then you go normally to your presentation.
A window. Huh. I can't find the file.
Okay, we still have a bit of time. Don't worry. We will wait for you.
So I'll share my entire screen. Google, so this is what, Google?
I recommend that you open your presentation and then you share a complete screen. So you have it already ready to use it.
I know, but it won't go.
So your presentation is open already.
Pardon?
Your presentation, your PPT is open already?
Yes.
Oh, okay, I see.
But I can't share the screen. It won't.
You have all the authorization. So,
I have no idea. I've never used this format before.
Don't worry. Just let me ask internally and I will let you know. Don't worry.
Okay. Here's the presentation. Here's this. Share screen.
Okay. It seems you have all the permission to share. So maybe you can send it, your slides, and we can share it for you. If it would be okay for you.
Yeah, sure. I should send them to
the email?
Fatima?
Yes, please. So we can
all right.
share it for you. Thank you so much.
Okay. I'll attach the file. Okay. I can't send it. It says the files are too large. Let me just try something here.
Okay. Yes, sure. We'll wait for you. Thank you.
I can't get past my privacy and security settings.
Oh, it seems it's in your computer. Okay.
Yeah.
Maybe I can create a drive. I send it to the chat and you can upload it there.
Okay, yeah, let's try that. I don't know. I've never used.
Yes, don't worry. We will solve this. We are on time. Perfect timing. Thank you.
So here's the chat. Okay.
Yeah, I will use the chat. I will just create a new drive for you. So you can put your material there. Just a second, please, to share it to you. Okay, let's see. Okay, just a second. So I will let you. I would put it in the chat, right? The Meet chat. Okay, so there is the drive. Okay, it's. Can you see it? See?
Yeah. All right.
Okay. Thank you.
All right. So let's see. Google Drive. Move a file. So I can just drop files. Okay, let me see. All right, it says it's uploading.
Okay, cool. Let me check.
All right, it says it's uploaded.
Oh, okay. I see. Thank you.
Okay.
So we will try to show it. Just wait a second, because we are going to introduce you first, and then we can start. Thank you so much.
Yep.
Good afternoon. On this occasion, we have the pleasure of introducing Dr. Bonnie Diddle, who is a senior researcher at the Varsity Blood Research Institute and a professor of microbiology and immunology at the Medical College of Wisconsin, USA. Dr. Diddle is a recognized immunologist for her research on the regulation of inflammation, particularly in multiple sclerosis and other autoimmune diseases. Dr. Diddle will present her talk title, B cells with low IgD expression, BDL, as a therapeutic strategy to increase the endogenous numbers of CD4 plus FOXP3 regulatory T cells. Dr. Diddle, welcome to this event. We appreciate your participation and support. Dr. Diddle, please remember that you have a maximum of 45 minutes for your presentation, after which we will have a 10 minutes question and answer session. We remind the audience in the room to keep their microphones and cameras off to avoid interruptions during the presentations. Questions shall be submitted via the chat during the talk and will be answered at the end. We also want to let you know that the link of the registration form will be shared on our social media channels during the presentation. Please, Dr. Diddle, you may begin. We are listening.
Great, thank you. So, okay, presenter mode. Okay, great. Hopefully everyone can see that. This is really exciting to be talking to researchers in Peru. I've always wanted to go to Peru. One of my favorite foods in the whole world to eat is Peruvian ceviche. And I eat it wherever I can find it. And someday I'm going to make it to Peru to eat it in your lovely country. So today I'm going to talk to you about a project that I've been working on for a very, very long time. And that is specifically how B cells function to regulate inflammatory immune responses. But since this is okay, I'm going to have to tell you to advance the slide. Okay, thank you. So since this is a symposium with a broad research interest, I thought I would give some background slides first on some components of immunology that might not be readable knowledge for everybody. So first of all, it's my research all sort of starts with autoimmunity. So autoimmunity is actually when your immune system attacks itself. And interestingly enough, there are literally over 100 different autoimmune diseases. Like autoimmunity can basically affect any organ and system in the body. And it's as a class of diseases that makes it the third most prevalent disease category. And it's very often chronic, and it's with you for life. And autoimmunity just kind of wears people down. And so they, can eventually become susceptible to other infections and things like that. And they often have premature deaths. There are no cures. And really the only universal therapy out there is really high dose steroids, which and then low dose steroids, right? As people are on them for a very long time. But it's really detrimental to body systems to be on steroids for extended periods of time. So the disease I'm mostly interested in is multiple sclerosis. And in that disease in particular, there are a lot of what we call disease modifying therapies. So in the case of MS and other diseases, it can kind of slow the progression of the disease, but they aren't cures. And so we still need, sort of new therapies that really can slow the progression of disease without some of the severe side effects that some of the disease-modifying therapies have. And so, people are out there trying to think of, can we think of a universal strategy in which to suppress, large classes of autoimmune diseases? Next slide, please. So when you think of the immune system, there's two different components. There's the innate immunity and adaptive immunity. Innate immunity is that immunity where your body can recognize a foreign microorganism, and that's based on structures on the microorganism, such as components of a bacterial cell wall, different kinds of DNA and viruses. And so this is very fast, and it can take hours to days. And your immune system is genetically hardwired to remember these structures, so that there is no immunological memory. You use the same mechanisms every single time you see the same flu virus, for example. Now, adaptive immunity is a little bit more sophisticated in the sense that it recognizes particular proteins, or what we call antigens, on the cell surface of pathogens. And so this is slow. It can take weeks to develop a full-fledged immune response to a new microorganism you haven't seen before. But the most important thing is you get memory. So then when you see that organism again, you can respond to it very, very quickly. That's sort of the concept of immunizations. You immunize. I've seen the antigen, I've driven an immune response to, say, influenza. When I get influenza, then my body already knows that I've seen this. So next slide, please. So how does all this work? So it has to work with antigen recognition. So there's three main players in this adaptive immune response. The first player is a dendritic cell. And that dendritic cell is the one that will come in contact, in this case, we'll stick with influenza. With influenza, it phagocytizes it into the cell and then chops it up into pieces that then can be presented on what are called MHC class 2 molecules, or class 1 molecules on the cell surface. And then T cells will see that peptide in the context of the MHC, and it can become activated. But at the same time, the B cells, if you look at the upper right, are recognizing the same virus, but with their B cell receptors, and they can detect the intact virus. And then the B cells become activated, and then they interact with the T cells, and then they crosstalk and communicate. And that's actually what then can drive production of antibodies, and also generates memory. So in autoimmune disease, you can have either T cells that drive the disease, or they go all the way to that antibody response, and then the antibodies can drive disease. So classic T cell mediated disease is multiple sclerosis. Antibody mediated diseases are like lupus, myasthenia gravis. Okay, next slide. So there are different kinds of T cells. And so T cells come out of the thymus as naive T cells. So you now have your helper puppies, and there are different kinds of T cells. There's CD4 positive T cells, which we call kind of helper T cells, who recognize MHC class 2. CD8 T cells have sort of innate cytotoxic ability. They recognize MHC class 1. But these CD4 T cells that come out of the thymus, they're like puppies. They're like naive. They aren't full grown yet. And when they when they're out, they're full grown, but they haven't really become what they're going to become yet. So they have to become activated in this process with interacting with the dendritic cells. And based on the various signals that they get, they're going to become different classes of CD4 T cells, which we call T helper T cells. For example, anyone who reads the immunology literature, you might have heard of T helper 1 or T helper 17. So they're similar, but they're different. So you can see the TH1 and TH17 dogs are similar, but they're not exactly the same, and they have different functions. Okay, next slide, please. Now the same thing is true of B cells. So now B cells mature in the bone marrow, and they enter the circulation as immature B cells. And then they actually go through multiple rounds of differentiation. And this differentiation is really well studied in the spleen. And so you can see that your mature B cell, it's like the kitten, and then it goes into the spleen and it has to mature. And one type of signal a B cell can get is a Notch 2 signal, and they become these marginal zone B cells. So that's the cat that's chasing the mouse. And the reason why that's what I use is because these marginal zone B cells stay in the marginal zone of the spleen. And I'm going to show you some slides later of what that actually looks like. And they're the first line of defense against bloodborne pathogens. So the marginal zone B cell is chasing the mouse pathogen. Now, sort of the default pathway and what most of the B cells are in the spleen are called follicular B cells. And that's largely, they reside within the B cell follicles. And again, I'll show you images of this. So the follicular B cells, when they become activated, they can rapidly divide. And so eventually what you have is a couple of kittens, and then you have more kittens. And then the B cell response matures and it gets better, and this B cell receptor gets better at binding to its antigen, and then you get lots of kittens. And most of the kittens are going to be like grayish color, because that's the B cell that responds the best to, in this case, we can say influenza. But there are also other B cells out there, like the gray kittens or the orange kittens, that will also be able to respond to the virus. But the gray kittens are going to be the best kittens because they're going to grow up and be able to attack the mice the best. Okay, next slide. So everyone knows about inflammation, right? It's a broad term. So inflammation is actually defined by pain, heat, redness, swelling, and loss of function. And of course, the immune system plays a huge role in inflammation, and both the innate and adaptive immune system have roles in inducing inflammation. So, when you get, like, let's say you get, we all get like stabbed by something in your hand, or you have a hangnail and you get that sort of inflammation and you get that sort of pus forming and it hurts and it's red. Well, in multiple sclerosis, can you imagine that's happening in your brain, right? This is not good, right? And so if you can see on the bottom left here, you can see all the purple nuclei there of cells surrounding a vessel there. And that's like immune cell infiltration. And that infiltration of the immune cells sort of sets off this cascade of a process that leads to demyelination. So if you look at the nerve on the upper right there, a nerve cell has the cell body and the axon, and wrapping the axon is myelin, and it's called the myelin sheath. And what happens is when the nerve impulses go, they track down the myelin sheath. So they flip from node to node, and it moves very quickly. But when the myelin sheath is no longer there, then that sort of nerve conductance is impeded, and then you can have neurological symptoms. And so if you look at demyelination, that's sort of the middle picture on the bottom, you can see that the myelin is in blue, and when there's no myelin, it's demyelinated. And if you had stained that lesion for immune cells, chances are there would be a lot of immune cells in that demyelinated area. And the way MS is mostly diagnosed is now you see those lesions in those demyelinated areas. And when you do an MRI, they show up as those white spots on the brain that are regions of where there's actually blood-brain barrier breakdown. And so those lesions, so people with MS can relapse and remit. So those lesions can kind of come and go and move around, and they can be anywhere in the central nervous system, from the optic nerve to the brain to the spinal cord. So the diseases you can imagine can be very neurologically disabling. All right, next slide. So if you were going to develop a universal therapy, what would that look like? And a lot of people have sort of honed in on these regulatory cells called T regulatory cells. In particular, they're a subset of those CD4 T cells I was talking about. So they're CD4 positive, and they express the transcription factor FOXP3, which is sort of the universal transcription factor that denotes them as a Treg, giving them Treg function. And so they're called T regulatory cells, or Tregs for short. And they really are essential for controlling immune responses to self and immune responses that prevent this sort of widespread chronic inflammation. And so humans, they first discovered this in humans who have this disease called IPEX, and they were found to have deficiencies in this transcription factor FOXP3. And then in mice, it's the same situation. If you knock out FOXP3, you can see that that poor mouse there is called the scurfy mouse, and that mouse doesn't have FOXP3. And so the way FOXP3, the way Tregs are mostly defined is they have FOXP3, and then oftentimes they have the high affinity IL-2 receptor as well, that's CD25, and there are other markers as well. And in the case of today's talk, I'm going to talk about a protein called GITR. You can see it there in orange, and it's highly expressed by Tregs, and when it is engaged, Tregs undergo proliferation. So there are a lot of companies and groups out there trying to find a way to use Tregs as a therapy to control autoimmunity and other inflammatory diseases, sort of like graft versus host disease. All right, next slide. So how are we going to contribute to this? So very early on in my career, I helped work on a study that showed that mice without B cells are unable to recover from this disease called experimental autoimmune encephalomyelitis, and it's the mouse model of multiple sclerosis. And if you see on the far left there, the mouse, it has a scoring system from 0 to 5. And so 0 is the mouse is completely healthy, and then at 1 they sort of get this, they begin to get this ascending paralysis. You get a tail weakness at 1, and then 2 you have total tail weakness, and they have sort of hind limb weakness like paresis, and then scale of 3 they have total hind limb paralysis. And at that point, if they get worse, the mice are euthanized for humane reasons. But you can see that curve there that we're showing. This is just any old EAE curve. And the mice sort of start to get disease around day 5 to 10, peaks around day 15, and then the mice kind of go, the inflammation is resolved, and depending on the strain of mice, the mice can sort of completely recover or sort of slightly recover, like shown in the graph on the left. And so if you deplete all your B cells with an antibody to CD20, or if you don't have B cells due to genetic disruption, what you actually have is, you can see here in the middle bar graphs, that we deplete all the B cells using flow cytometry. You can see on the bar graphs, there's no B cells. And then you reduce your T regulatory cells by like 50%. And then, I'm sorry, this slide got cut off, but if you induce EAE in these mice without B cells, then the mice can't recover. So this early data suggested that there is a B cell out there. You can call it a regulatory B cell if you want. It's not a term I typically use. It's just it's regulatory function. And so then we had to figure out what B cell was this. All right, next slide. And I won't go into all the details because it's published and it's years and years worth of work. But eventually what we found is that when you then you could also partially deplete B cells as well. And so we found that, going back to the different kinds of B cells, follicular B cells, marginal zone B cells, that the BDLs, we call them BDLs because we found out that they have low levels of the immunoglobulin receptor IgD. So when B cells come out of the bone marrow, remember I told you they came out as immature, and then they mature into these follicular B cells, but they're still naive, right? So they still need to grow up if they're going to produce antibodies. So when they're naive, they express a immunoglobulin called IgM and also IgD. But for some reason that we have not, we don't understand yet, is BDLs have very low or no levels of IgD. And then I'm showing you a flow cytometry gating scheme on how you identify various populations of B cells. So if you gate on B220 positive B cells, sort of in that yellow circle is where these follicular B cells reside. And then you can move to the next panel that's labeled B there, where now you separate them again on CD23 and CD93. CD93 goes down with an expression as B cells mature. So then you take that gate, and then you look at IgD, and you gate on the IgD lows. And then we found another marker, which I'm not going to have time to show you how we discovered it, for BDLs called CD24, and they're CD24 high. So at the very end, so you can watch how the BDLs were phenotype across the gray shading. So those are BDLs. All right, next slide, please. And how do we know they're BDLs? Because we adoptively transfer them into MUM T mice, or mice that are B cell deficient due to a genetic disruption of the IgM heavy chain. And so if you look at the graphs on the left, so wild type mice have, about 3 million Tregs in their spleen. If you don't have B cells, that's immune T, it's about 50% lower. If you adaptively transfer total B cells, you get this Treg expansion, and you get Treg levels about wild type level. If you transfer BDLs, you also get Treg expansion. But if you transfer that follicular B cell population, you don't. And then high numbers of Tregs correlates to recovery from EAE in the graph on the right. And you can see if you look at the stars, then only the transfer of the BDLs will lead to recovery from EAE, and the transfer of the other two populations of B cells, folliculars and marginal zones, don't lead to recovery. So that is our evidence that BDLs are regulating EAE, promoting recovery via expansion of Tregs. All right, next slide. So one question that we were very interested in is, where do these BDLs interact with Tregs in the spleen? And actually, my graduate student who did this work, Angela Baltrami, just defended her thesis earlier this week. So this is really nice data. And so this is just a cartoon that she made showing you what the splenic architecture is in the, regarding immune cells. So if you look in this upper square where it's all red, that's what's called the red cell. The red pulp, where the red blood cells are. And then the purple is this area called the marginal zone, which is where the marginal zone B cells are. And then in the middle, you can see the green, the kind of fluorescent yellow green is the T cell zone. So that's where the T cells are. And then in the blue are the B cell follicles, where the B cells are. And then in particular, we're very interested in the green area, which we call the TB boundary. And so we hypothesized that at the TB boundary is where the BDLs and the Tregs would be interacting. All right, next slide. So this actually was a very difficult thing to do, because the BDLs don't have any cell surface marker that you can stain for and go, yes, I'm a BDL. So we had to develop a strategy we use IgM and IgD to do this. But first on the left, I'll show you this sort of splenic architecture so you can see it for yourself. So you can see it in the middle and sort of in the red. Sorry, it's not showing up very well here, but that's the T cell zone. And then you can sort of see the little blips or ears circulating like clover, I guess like a little clover around the T cell zone. Those are the B cell follicles. And then the white is the marginal zone. So you can see that there's always this area where the B cells and the T cells are together and are interacting, or have the potential to interact. So how do we find BDLs? Well, we know they're IgM positive, so that's in the teal, and the arrows are showing IgM positive B cells, but then they don't express IgD. So you can see if you look at the arrows, there are B cells that express IgM, but not IgD, so we call those BDLs. And then the Tregs are really easy to find because they're CD4 positive in red and FOXP3 in the nucleus in green. So this is our strategy in which to find BDLs, and then Angela's task was to map where they interact and how they interact. Okay, next slide. So we also were interested in this proliferation. So if all our data showed that BDLs interact with Tregs and induce their proliferation and their expansion. So this is just one more higher magnification view of these Tregs. So again, Tregs are phenotyped by on the histogram on the the flow cytometry plot on the upper left is CD4 versus FOXP3. So you gate on the FOXP3 positive cells. And then if you ask how many of them are proliferating, we did that by staining for Ki67, which is a cell cycle marker. So it sort of starts to come up when the cells proliferate. It's highest when they're in that part where they're actually dividing, and then it comes back down. So you always have an intermediate population of Ki67, but the high population are the ones that are actively proliferating. And you can see that in our histology where the red, the CD4s, and then we can see that they're FOXP3, and then the Ki67 in the blue. And so you can measure the intensity of that Ki67 staining, and then you can sort of map if the BDLs are next to Tregs that are proliferating or Tregs that are not proliferating. Okay, next slide. So this is how Angela did this. She used Fiji ImageJ and a LabKit plugin, so that what she did was she was able to find the Tregs in the far left. And then she did a IgM-IgD segmentation. And then she wanted to just find the IgD lows, and then she could overlay them all together on the far right. Okay, next slide. And so when she did that, so they're called Treg neighbors. So if a Treg, so when you look at just the Tregs, you ask how many of them are neighboring a BDL, the percentage, versus how many are not. And it's interesting that Tregs like to be next to the BDL. So you can see it's a ratio of, 60 to 40. So they like being next to the BDLs. And if they are next to the BDLs, are they proliferating? And so that is showing in the right graph. So you're showing, are you, if you're neighboring a BDL or not, do you have a high, a Ki67 signal? And here the data shows that if you're next to a BDL, you're more likely to be undergoing proliferation. So this is really good. It fits our hypothesis that, B-cell BDLs interact with Tregs in the spleen and induces their proliferation. All right, next slide. So they kind of provide a proof of concept that you could develop and adopt a cell therapy to use BDLs to drive Treg expansion. So next slide. So this is our proof of concept. So this is WeHi 231 is a mouse B-cell line. And if you phenotype it, it kind of looks like a BDL. So if you look at it, oh, sorry. This is my Mac to a PC conversion here. So I'm going to have to, so the left, no, I have to remember what they are. So the left graph, I know this is probably IgM. Yeah, the left graph, the leftmost histograms are IgM. Then it's IgD, and so you can see that BDLs have low levels of IgD, GITR ligand then, and the other ones are not as important. But all in all, phenotypically, they look a little bit like BDLs. And so to test their regulatory activity, first, this work was done by Zavar Hadja, and so she had to sort of, since it's a cell line, she had to sort of irradiate them and go at what level do they stay alive, but you radiate them so that they can't proliferate in the mice and form lymphomas and such, that they'll stay alive but they aren't proliferating. So she found that dose of irradiation, and then she transferred them into a different mouse, a bulb C mouse, but this mouse is also rendered B-cell deficient with a knockout called JH. That's a knockout of the J region of the heavy chain of IgM. And so the same thing happened. So if you have the bulb C mice, you can see the bar graphs on the far right, you have Tregs. If the bulb C mice don't have Tregs, they have reduced numbers, about 50% of Tregs. And if you give them total B cells, you get your Tregs back. And if you give them these WeHi cells, you also get Tregs back. So we didn't do anything to these WeHi cells, actually, and besides grow them. And it just kind of shows that, B cells can induce Treg proliferation. And studies that I don't have time to show you today that Angela also did was sort of figure out chemotactic factors that are required to get BDLs to go to where they need to go to interact with the T cells in the spleen. And I won't have time to talk about those today, but hopefully those studies will be published soon. All right, next slide. So that was one strategy, sort of develop a prototype of BDL that you could adoptively transfer into people to increase endogenous Treg levels. But as we were thinking about these strategies, we came up with another one, and this one is bone marrow transplantation. So next slide, please. So first we had to convince ourselves that there are BDLs in the human spleen. And these experiments are published, and we phenotype them very similarly. So you phenotype first on CD19, And then CD20 is also an antibody that's a pan B cell marker like CD19. So in the dot plot, the middle dot plot, you can see that we gated on CD20 and CD24 low. And this tripped us up for a long time because we knew about the CD24 low in humans before we knew about the CD25 in mice. And it just goes to show, mice and humans aren't the same. And CD24, like I told you, like CD93, also comes down during differentiation of B cells in the spleen. And for whatever reason, in the spleen of humans, that kinetics is a little bit slower in the mice. But then if you gate on the IgD lows versus the IgD highs, you can see that we did a CFSE cell proliferation assay. So that what that is, when you label the outside of the cell, you basically label all the cell membranes in the cell. So the idea is when the cell divides, that fluorescence, will divide in half. And so when you look at it on a flow cytometer, the peak will shift by 50%, by half. And then the more it shifts to the left, the more it proliferated. You can see that there's always some background proliferation when you just put Tregs in the system, because the Tregs are being stimulated with anti-CD3. And then you have total B cells and you get about the same level of background proliferation. But if you put in these IgD lows, you increase that level of proliferation. So from this, I know it's a little crude, but from this we sort of go, okay, I think there are human BDLs. All right, next slide. And so then we wanted to go, okay, there's human BDL, so let's keep going on with our therapies. And so how are we going to really get these BDLs to give us high numbers of Tregs? And so we didn't develop this mouse, but it's a GITR-like, no, sorry, I'll go on to that next. So in this case, we wanted to know what the mechanism is. And we did look at a lot of potential mechanisms, and we actually, it's always the last one, right? That's the answer. So in this case, we got a blocking antibody to GITR ligand so that we could block the GITR ligand on B cells before we did the adaptive transfers. And so if you look in the bar graph on the far left, again, if you give them BDLs, you get your Tregs back. If you block GITR ligand on the BDLs before they're transferred, you do not get the increase in Tregs. And then obviously the follicular B cells are the controls. So and then that correlates directly into EAE. So if you block the GITR ligand on the B cells, they can't recover from EAE. And then we're also interested in how long the increase in Tregs lasts. And the bar graph on the far right is day 30 of EAE. And you can see that at least for 30 days, this increase in BDLs occurs. And so here's just the little cartoon on the right showing that, GITR and GITR ligand would engage with each other and induce Treg proliferation. And that supports really well with our data in the spleen, showing that the Tregs that are proliferating are adjacent or neighboring BDLs. All right, next slide. But then how can you, wow, this is really a mess. So how can you sort of use what we already know to develop a therapeutic strategy? And this mouse is a GITR ligand transgenic mouse, in that it only overexpresses GITR ligand in B cells. And if you can see in the histograms, you can sort of convince yourself that if you look left to right in the top three histograms, that there is a shift, and the GITR ligand transgenic mice do have higher levels of GITR ligand in all the populations of B cells. And these mice have higher numbers of Tregs, so I'm glad I labeled the top. So you can see that the first bar graph is total splenocytes. So you have more splenocytes across the board. It's not just Tregs. And then the next bar graph, the non-Tregs, is really a CD4 positive T cells to show you that this overexpressing GITR ligand in B cells, for some reason, homeostatically expands all of the lymphocytes and myeloid cells too, which I won't have time to show you. And most importantly for us, you have this large increase in Tregs. And then if you look at the percentage of Tregs within the CD4 subset, that is also greatly increased. And that is what is most exciting to us. because there's kind of this concept of it's the ratio between Tregs and cells that it's suppressing that's really important. So if you're trying to suppress CD4 positive T cells in this case, because they're the effector cells in multiple sclerosis, if you have more of them, than you would have in a situation where you don't have GITR ligand overexpression in B cells, then in theory you should suppress better. And when you induce EAE in the GITR ligand transgenic mice, you can see that it is attenuated. Those are the dark circles in the EAE graph on the left. It's not as great as you would think it would be. I always thought that was really interesting. And then we always do our statistics using area under the curve, and that's shown in the bar graph on the right. And it's statistically different. All right, next slide. So then we wanted to ask, can we do bone marrow transplantation? So this is a straightforward experiment. And the control is wild type bone marrow into wild type. And then the experimental group is GITR ligand bone marrow transplant into wild type. And then we wait 10 to 12 weeks and we induce EAE. And you can see now that EAE is almost completely suppressed or attenuated. And so that is really, really exciting. And then we also wanted to see how many Tregs we have on day 30, and that's in the bar graphs on the right. And so the controls are wild type and the wild type are GITR ligand, the transgenic into the wild type without EAE, and then the ones with EAE. And you can see the bar that the levels of Tregs are exactly the same. And so the EAE didn't impact Treg numbers. So that was really exciting to us. And so we're sort of pursuing this strategy as we move forward to try and develop the therapy. All right, next slide. So, all right, next slide. Sorry, I should have taken that slide out. Okay, so we are convinced, we hope, that there are human BDLs. So how do you find them? In a mouse, it's a lot easier because mice are living in a cage. They're really not exposed to many things. Most of their T cells and B cells are naive. But in humans, that's not the case. So if you remember, our B cells are IgM positive, IgD low. And if you look at the B cell literature, there are at least 10 populations of B cells, including the BDLs, that have that phenotype. So you have to use really sort of high-level phenotyping strategies, and usually you do this by flow cytometry. And so, again, if you use combinations of CD21, CD27, CD24, and CD38. And so of course, you always start with CD19 positive cells, and then we're most interested in IgD. So if you segregate out the CD27 by IgD, the 27 is sort of a marker that we sort of say are the memory B cells. It's not an exclusive marker, but if they express CD27, you would say their memory. And then you can see that we do have this population of IgD low positive cells in quadrant 4 on the far left of the first dot plot. And then we're like, okay, we have to take these cells and separate them out even more. So then you can sort of look at CD24 and CD38, and what we really are interested in are the naive B cells. And you can see that circle in the middle are what we consider the naive B cells. So you can move those forward. All right, next slide. So to do this, we developed a multi-step strategy to characterize these IgM positive, IgD low B cells. And we use a combination of flow cytometry, as I showed you, site-seq, and single-cell RNA-seq. And so if you see on the left, we purify our we purified our CD19 positive, IgM positive, IgD low to negative B cells by FACS sorting. And then when you do that, so this population that we were interested in, of course, is the population on the far left or the top. Yeah, sorry, that you can see the two circles that are IgM, the one on the top is IgM positive, IgD negative. And you can kind of move that forward. And then these were labeled with site-seq antibodies. So that means you're putting antibodies on the cell surface that have oligo tags that can be amplified. And then we use our 4 markers, CD21, 27, 24, and 38. And then we also were able to generate UMAPs, and there were 3 donors. So you can see that in the UMAPs, when you combine all the donors together, there's good overlap. And then when you look at the cell clusters, you actually then are able to see there's activated memory, naive, resting memory, and transitional B cells that we can detect. All right, next slide. And then what we found that was very surprising is that about 50% of the B cells expressed, even though these were IgM positive, they shouldn't have IgG transcripts that are, IgG or anything else. But about 50% of them did. And so you can see in the upper graph that you can see all the ones sort of in the reddish, I guess, salmon is, that they have IgMD and something else. And it's only the green ones that have IgM and or D only. And so we took all the B cells out that had some other Ig besides IgM and IgD. And then you can see with down below then in the teal and the salmon that you have true and false. So the true are the blue and those were moved forward. And then we use our site-seq markers to go, hey, can we recapitulate this flow cytometry gating that I showed you in the very beginning? And we were able to do that. All right, next slide. And even when we did all of that, and you narrow it down, you still have 3 populations of B cells. So we have an immature population, a naive population, and an activated population. And so we're hypothesizing that our BDLs are in this naive population. And then what you can do is once you've narrowed it down to these are the cells I'm interested in, then we can do you can show the heat maps to show that the 3 populations are different. And then you can start looking at their transcriptions. And so we have done all of that. And this actually, I should have told you, was actually done on peripheral blood to develop the pipeline. And now we're doing the same thing in the spleen, and we have the cDNA libraries and their sequence, and we have already identified some targets that we want to look at so that we can better identify human VDLs and try to understand how they function better. So that's where we are on with that project. All right, next slide. So here's my lab. The people in the left in black are the people who were in my lab that really contributed a lot to this project. Monica Mann was my first graduate student. Avijit Ray and Mohammed Khalili were postdocs. I already talked about Angela Baltrame. Nate Meinhardt is doing the human studies. Cody Gursky is the best lab manager on the planet, and he helps me run my lab. And yeah, so those are the main people working on this project that have contributed, and there are, all my other collaborators as well. I think I have one more slide, I'm hoping. No, guess not. Okay. All right, that's it. I'm sorry, those just go back to my, So I wanted to put a slide in there, but I guess in all my saving and all my troubles trying to get this to go, I had a slide in there that I wanted to encourage people to publish in a journal called ImmunoHorizons that I'm editor-in-chief of. And this is an open access journal that's published by the, it's a journal for the American Association of Immunologists. So it's American Association of Immunologists. So anyone can join this. And we have, it's published by Oxford University Press, and it has agreements with a number of research institutions around the world, actually, that can sort of give waivers for the open access fees. So if anyone is interested in ImmunoHorizons or publishing in ImmunoHorizons, please let me know and contact me. Now, if I should be on time, I hope, and then I'd love to answer questions.
Thank you very much for your presentation, Dr. Diddle. Now, Dr. Llanos will proceed to deliver the questions from the audience.
Thank you, Diego. Good afternoon, Dr. Diddle. It's our pleasure to receive you and to listen to your wonderful talk. So I have some questions for you, and let's start. The first question is, Dr. Diddle, what cellular mechanisms has been proposed to explain how B cells with low IgD expression can enhance the endogenous activity of the immune system?
Well, I wouldn't say I've looked at the endogenous activity of the immune system, because if you follow B cell development, and when they go into the spleen, and I didn't go into all of this, they go from the immature, and then they go through transitional stages, transitional 1 and transitional 2. And then it's kind of like the BDLs, and then it's the folliculars. If you look at the developmental trajectories that we got by single cell RNA-seq. And so I think, Angela's data showed that they're in this area at the T-B cell zone. So they're not really, and they're not in the marginal zone, and they're not in the follicles. So they're not really in a place where they would really be engaging antigen. So I don't think they do much else besides engage with Tregs. And whether or not they have the ability to go into the follicles and become classic follicular B cells is a question we would like to answer, but it's very difficult to do those type of fate mapping studies on B cells because they don't have definitive markers by which you can track them.
Okay, let's go with the following question. Okay, let's see. Dr. Diddle, considering the differences between innate and adaptive immunity, how do these two branches of the immune system influence the regulation of IgD low B cells that you study as a potential therapeutic strategy?
Well, I don't think the innate immune system has anything to do with it, really, with B cell development. B cell development sort of occurs, it begins in the bone marrow, of course, with stem cells. And as they receive certain signals, they have to decide whether or not to go down, myeloid lineage, the lymphoid lineage. And then there's signals that will determine whether the lymphoid progenitor becomes a T cell or becomes a B cell. And then the B cell goes through its maturation. And so it's kind of a process that sort of happens. I mean, it's obviously regular, obviously it needs certain cytokines and certain signals it gets from stromal cells and other cells in the bone marrow, but it sort of happens. And then marginal zone B cells in particular are the ones that have to receive a certain signal to become marginal zone B cells, and that's Notch2. And its ligand is delta-like 1, and there will be stromal cells or some cell type in the it's non-hematopoietic cell in the spleen that delivers that signal. So it's sort of a process that happens that I wouldn't say necessarily is driven by innate or adaptive immunity. It's a developmental process.
Okay, I see. Do we have other question? Another one? So, Dr. Diddle, how do BDL cells contribute to the induction or expansion of the CD4 plus FOXP3 plus regulatory T cells?
Yeah, so we don't have any evidence that it's anything other than the GITR ligand on the B cells interacting with GITR on the T cells. We've looked at other mechanisms and we haven't found any. And so you also get an expansion of conventional CD4s as well, because they can have a GITR as well and CD8s as well. But it's something I don't understand, and it's still perplexing to me as to why you would get an increase in all the cells. So it's somehow like the cells are hardwired to have a homeostatic balance. Because when you look at the mice and you look at the ratios of, the CD4s, the CD8s, we looked at myeloid cells kind of grossly with CD11B and asked, what are the ratios of those different kinds of cells? And they're exactly the same, whether you're a wild type mouse or your GITR ligand transgenic mouse. So it's some kind of hardwired homeostasis that the cells say, I need this many B cells, I need this many T cells. And I thought that was very fascinating.
Okay, we agree. Everything is fascinating what you show us. So I have other question. How do you envision integrating BDL cells-based therapy into the immunotherapy landscape for autoimmune diseases? For example, could it be combined synergistically with approaches such as low dose of IL-2 administration to expand the Tregs, or with CAR Tregs cell therapies, creating a more robust and long-lasting regulatory ecosystem?
Sure. if we could figure out how to make a universal BDL, which we have thought a lot about, but I didn't really talk about it much here. Yeah, of course, you could give it with other therapies and keep your Treg levels high. But the strategy that we're mostly going after at the moment is bone marrow transplantation. And to do that as a proof of concept, we did it in EAE, but you have to convince physicians that they should transplant their MS patients. So we decided to move to a system of graft versus host disease after heterogeneic bone marrow transplant. And because those patients that get, they're mostly getting transplants for leukemias and things like that, they're getting transplants anyway. And so that way we could incorporate our BDL therapy. So you would use like a lentiviral vector. That's the strategy to, transduce the stem cells. And then when the B cells develop, they'll overexpress GITR ligand. So that's the idea. And to sort of start graft versus host disease, because that is also a disease that a lot of people who are developing Treg therapies are targeting as well. And then, prove it that it works and then move to autoimmunity. Yeah.
Okay. Okay. I guess I have another question. Let me check. People is very enthusiastic. So I have this question. Treg plasticity presents a therapeutic challenge. When Tregs are induced or expanded by BDL cells, are they stable and functional long-term in an inflammatory bioenvironment? Or is there a risk that they will lose the FOXP3 and become effector cells, potentially exacerbating the disease?
Yeah, I kind of get that kind of question a lot because these are naive mice. I mean, once they get EAE, it's not clear if any of the Tregs are inducible or not. But when you do the experiments in a naive mice, I would think most of them are natural Tregs where they come out of the thymus. And yes, people have shown that antigen-specific Tregs seem to be better at suppressing. But so, there are some markers where you can say where they're natural Tregs or induced Tregs, but they're not totally specific. You know what I mean? Where this is this Treg was induced. And so we haven't really studied that. People always tell me I should study it. And so we are. And so we are doing it by single cell RNA-seq. And we're just starting by looking at the Tregs. What do the Tregs look like in the spleen when you have B cells and when you don't have B cells? Can you find a difference? And then, of course, you can look for the proliferating Tregs if you want. But even that's hard to do, right? Because there's Tregs all over the T cell zone. So how do we isolate and localize the Tregs that are just next to the BDLs? And to really do that, I'd have to do spatial transcriptomics. And it's just not something that would be easy to do. You could find the Tregs. I mean, with spatial, you can find the Tregs. Yeah, it would be hard. Maybe it could be done, but it would be really hard. And I think that's the way you'd have to do it and go, are the Tregs that are adjacent to the BDLs different than Tregs that are someplace else in the T cell zone?
Okay. So thank you very much. Now I'm going to ask Diego to finish delivering you our gratitude for your participation. Thank you very much.
We thank Dr. Diddle for her excellent presentation, which I am sure has been of great benefit to everyone. Thank you for sharing your knowledge and experience in cell and molecular biology. We also thank this morning's participants for their attendance.
Thank you very much. It's my pleasure.
You're welcome.
Hemos concluido con las ponencias de manera muy satisfactoria en estos 2 días de desarrollo del 6º Simposio Internacional de Biología Celular y Molecular 2025, con la gratificación que produce el recibir conocimientos en tópicos de biología celular y molecular de sus propios autores, con reconocida trayectoria científica. Además de cumplir con el deber de difundir el conocimiento científico y de crear un espacio que fomente la discusión de temas actuales de gran relevancia dentro del campo molecular y celular. Para cerrar oficialmente este evento académico, invitamos a la doctora Angelita Cabrera de Cipriano, directora de la Escuela Profesional de Ciencias Biológicas de la Facultad de Ciencias Biológicas de la Universidad Nacional de Trujillo, Perú, para que nos brinde el discurso de clausura del sexto Simposio Internacional de Biología Celular y Molecular 2025. Le escuchamos, doctora.
Muy buenas tardes, distinguidos ponentes, docentes, queridos estudiantes y apreciados invitados. Es un honor tener esta oportunidad de comunicarme con ustedes y participar de este magnífico sexto Simposio Internacional de Biología Celular y Molecular. Un espacio académico que gracias al esfuerzo y compromiso de la promoción 62 de la Escuela de Ciencias Biológicas ha logrado reunirnos y transmitir y adquirir conocimiento, innovación y una profunda pasión por la ciencia. Durante estos días hemos compartido investigaciones, experiencias y reflexiones que enriquecen no solo nuestra formación profesional, sino también nuestra visión sobre los desafíos y oportunidades que nuestros estudiantes en el campo de la biología moderna se nos presenta. Cada conferencia y cada diálogo nos ha recordado que la ciencia avanza cuando existe trabajo colaborativo, cuando hay curiosidad, con el firme deseo de comprender los procesos de la vida. Quiero expresar mi reconocimiento a los organizadores, quienes han demostrado liderazgo, responsabilidad y un admirable espíritu académico. El esfuerzo, la dedicación ha hecho posible un evento de este nivel, que fortalece la proyección científica de nuestra escuela y reafirma el compromiso de nuestras futuras generaciones de biólogos. Agradezco también a los investigadores nacionales e internacionales que nos han brindado su tiempo y su sabiduría. Sus aportes inspiran a nuestros estudiantes a seguir explorando, preguntando, construyendo este campo de la ciencia con rigor y ética. Con la satisfacción de haber cumplido, declaramos oficialmente clausurado este sexto Simposio Internacional de Biología Celular y Molecular. Que lo aprendido aquí sea el punto de partida para nuestras ideas, nuevos proyectos y nuevas oportunidades de crecimiento y desarrollo. Muchas gracias.
Agradecemos a la doctora Cabrera por su participación. De hecho, agradecemos a todos los investigadores que han realizado sus presentaciones y que han dado realce científico a este evento. Por tanto, agradecemos al doctor José Morgado Díaz, a la doctora María Marzolo, a la doctora Irene Consuelo Julca Chávez, al doctor Harvey Lodish, a la doctora Karen Lorena Chauca Espinosa, a la doctora Hilary Kohler, al doctor Luis Felipe Jiménez García, al magíster Lionel Germán Acosta Campos y a la doctora Bonnie Diddle. Agradecemos a todas las personas que se encuentran con nosotros en las diferentes plataformas de transmisión por la atención brindada y de manera muy especial expresamos nuestro agradecimiento a los ponentes, quienes tienen nuestro reconocimiento por su apoyo en la divulgación del conocimiento científico de manera gratuita, el cual garantizó el éxito de este evento. Asimismo, felicitar a los intérpretes por su extraordinario trabajo y su espíritu de colaboración. Les informamos que la entrega de certificados está supeditada a su participación activa enviando 7 de 9 formularios que evidencian su asistencia a las diferentes ponencias de este evento gratuito. Toda información sobre ello la estaremos compartiendo en la cuenta oficial de Facebook, CIT's International Symposium on Cell and Molecular Biology. Finalmente, les invitamos a seguir acompañándonos en las próximas ediciones de este evento. Muchas gracias a todos.